for continuously without any rest, you are organizing the webinars, even this pandemic situation. And today's topic, research methodology, is most important thing for any scientist. So especially the Ayurvedic faculty, it needs more scientific approach, and students, especially scientists, young scholars, they need proper guidance, proper knowledge of how to research and the things, and how to brought the knowledge of ancient science to the scientific fraternity. So nowadays, everybody needs evidence-based studies, evidence-based. So that's why the research methodology, everybody should know each and everything A to Z about the research methodology, and they have to adopt it to convince the scientific fraternity. That's what I feel. And so this topic is very good. And I never met the speakers. I'm very happy to meet them today. And I request all of you to know about our Ministry of Ayush and our CCRS, Central Council for Research in Ayurvedic Sciences, and as well as Central Research Institute of Ayurveda in Kolkata. So being the head of this institute, we are having several activities of research, clinical, and with all the allied subjects research also is going on in our institute. So please go through our websites of council and ministry, and every student should know about the research councils, especially in the which are in the Ministry of Ayush of central government. So whenever I go to any college or whenever I ask any student, they doesn't know about the councils, research councils, what they are acting, what they are doing in the research, though everybody should be aware, and that is our responsibility to bring awareness about the Ministry of Ayush and research councils and what the research is going on in our central government. So I wish all the best, and I welcome all the speakers. Now I declare that this session is open now. Thank you, sir. Thank you, Sharmaji, and welcome to all the speakers and participants.
Thank you. Thank you. Thank you, Dr. Prasad. I would like to invite now our first speaker, Professor Suhas Kumar Shetty from SDM College, Hassan. He is head of the Department of Manas Roga, and he is Dean of Academics also. He has various research awards, webinars in his account. He is the dignitary in South for Ayurveda, not in South, for all over internationally. I would like to request Dr. Suhas Kumar Shetty ji to guide us on research methodology. Welcome, welcome, Professor Suhas Kumar Shetty ji. This program is live on YouTube and you can circulate it all. Welcome, sir.
Can you just allow me to share the slides? It says the host disabled participant screen sharing. So you need to make me co-host.
I'm making. This is Zoom. I am habitual on Webex. This is Zoom of Ayush Darpan Foundation. Just a thought. I have made you co-host.
Yes, sir. So the slides are visible?
So it's visible.
Yes.
Okay.
Good afternoon to all of you. On behalf of the Independent Research Ethics Society and Ayush Darpan Foundation, I thank all the organizers, especially the senior guiding force of this webinar, Dr. Pawan Sharma sir, Dr. Vishnu Prasad Sharma sir, and all the faculties and senior persons who have joined here. It's always a great pleasure to be associated with IREC and witnessing various very unique programs and unique webinars being conducted by the organization. And it's always a pleasure being a part of this particular program. And I'm very happy to be a part of one of the area where I have been very passionate about for the last 2 decades in the area of the research methodology and also the medical statistics. So being the Dean of the Research and Development in the institution and coordinating various research activities, let me just share a few of the very important concepts related with research methods and research methodologies. So the topic that I have chosen for the presentation for the next maybe for 20-25 minutes would be on, because whenever we talk about research methodology, the most important thing that comes to our mind is about the research designs, or what we commonly call as the study designs. So what should be the various study designs that we can use apart from the normal interventional or experimental study designs in Ayurvedic or any of the Ayush streams? So this will be probably the focus of my presentation of what are other newer study designs that one should know about in cases of the research that to be undertaken in the field of Ayurveda and other related traditional system of medicines. So I'll be focusing on basically what do you mean by research designs and how do you select a research design. I'll be highlighting and giving some examples of some of the common research designs and also talking briefly about their functional features, their advantages, disadvantages, pros, cons, strength, and limitations with some examples. So whenever we talk about research designs or when we talk about the research or the study designs, I always feel that it's like building your dream house. Like you want to build a house, and that is how you need to have a proper planning. You need to have the proper site, then you need to have the proper budget. So whenever you want to plan any studies, any research studies, any clinical trials, it's very important that you should have a clear protocol, a clear plan for your studies. So similarly, whenever you want to do any type of studies, so you always start with a research question, and then you have then you formulate a research objectives, then you have, and to answer these objectives, you should take up the appropriate study designs. So knowing the different types of study designs is very important for any research activities. So let's just see what are the common types of research designs that we can think of. The different types of research that we are mentioning in Ayurveda includes the Shastra Anusandhana, which includes the literary and the fundamental research, because this is a very, very vital area that we need a lot of research activities. Especially in our institution also, we have a separate manuscript laboratory where we have trying to validate, upgrade, edit, digitalize various types of digital manuscripts that we have collected in and around our place. We have more than 1,800 manuscripts on which we are studying at present and trying to digitalize these manuscripts. And also one more very, very important areas of research is the fundamental research, because Ayurveda strongly believes in various fundamental concepts like dosha, dhatu, mala, agni, srotas. So these are the areas where a lot of research need to be carried out. And also, we also need to develop some important diagnostic and assessment parameters as per Ayurvedic guidelines. Suppose you want to assess agni, ama, or srotas. So there should be some basic parameter. I think that is where probably we need to take the help of the basic science like physiology, biochemistry, pathology, physiology. So these basic science should be utilized, and probably if we can study on these basic parameters, I think it's a very important area that we need to focus upon. Of course, on the drug research, especially focusing on the Ayurvedic concepts of Rasa Panchaka, one should do the various steps of drug research are very, very important. Then the pharmaceutical research, where you are trying to standardize, looking about the safety and efficacy of the various single drugs, herbo-mineral formulations, herbal combinations, etc. And of course, the focus of this webinar will be on the clinical research. So whenever we talk about any research, especially when it comes to the clinical research, we always start with a research statement. Like, for example, we say that Vishado Rogavardhananam Shresthaha or Saumanasya Garbhadharanam Shresthaha. So these are some of the few of the statements that you find in case of a Ayurvedic literature. And from this, we derive a research question, which need to be assessed. And whenever you're talking about selecting a research question, there are a few guidelines, like you use the finer guidelines, or what also called as the finger guidelines, which talks about the feasibility, interesting, novelty, ethical, the reproducibility, relevance, scalability, etc. And whenever you want to do any of the clinical studies, we always follow a PICO, like the population, intervention, comparison, and the outcome. And also you need to fix your specific objectives, where the specific aims and objectives should be specified, which are specific, measurable, achievable, relevant, and time bound. And for answering these research questions, you should have a proper research designs. And this research design can be of two types: the study designs and the statistical designs. And then you frame your research hypothesis. You use the appropriate methodology and collect the data and analyze them and draw the final conclusion. So this is probably what you do in most of the clinical research. So to answer these research objectives or research questions, you need to select the most appropriate study designs. And the study designs are defined as the structured approach to address the research questions. And these are based on the population, what you want to measure, whether it is a nominal data or whether it's a categorical data, a numerical data, whether it's a quantitative data, qualitative data, primary data. So all this need to be understood whenever you want to address the issues. And then you should understand how you're going to analyze these data. So whether it will be the qualitative analysis, quantitative analysis, thematic analysis, etc. So if you look at the basic classification of the studies, we can classify it as descriptive studies and the analytical studies. Basically, descriptive studies have a comparators. Basically, you do not have a control group or a standard group in case of a descriptive statistics. The survey study or the qualitative study designs. There is some disturbance in the background.
I would like to request all audience kindly mute your voice, please. Thank you.
Yeah. And regarding the qualitative research, because we have been morely focusing on the quantitative research, and sometimes qualitative research is equally important, especially when it comes to the traditional system of medicine like Ayurveda. Like you should go about with the ethnographical studies, phenomenological studies. See, basically, suppose you want to know about the experience of something or the practice of Ayurveda, or you want to know about the KAP studies, the knowledge, attitude, and the perception studies, or you want to know about how people are perceiving about Ayurveda. So all such qualitative research, suppose somebody undergoes the therapies, Panchakarma therapies, or Abhyanga, or Basti, what is their experience about this? All these are very, very important. And it is not just the quantitative studies, the qualitative studies, or sometimes even the mixed methods of the qualitative and quantitative methods are equally important in Ayurvedic research. And the analytical studies can be classified as experimental studies, which has randomized and non-randomized, and randomized can be of the parallel group or the crossover groups. Whereas the analytical observational studies could include cohort studies, cross-sectional studies, and the case control studies. So when do we need the qualitative research methods? So when you cannot quantify something, but it is more of a attribute or quality or perception, opinion, attitudes, experiences, behaviors, then you need the qualitative methods. And suppose you want to have the understand in the real settings or in the real time, and you want to know about the roles, processes, and groups. And especially it's about basic understanding. And what are the various methods that we commonly use for qualitative methods? It includes the qualitative study method, the phenomenological studies. Like phenomenological studies is basically to understand why people behave in the way that they behave. Like why people are acting differently, or why the people are trying to behave in different aspects. Then you have the case studies, then ethnographical studies. Suppose you want to know about the culture, or you want to know about the different aspects of the studies, or you want to do narrative studies, etc. Then you need different aspects of a narrative analysis. So coming to the quantitative research, see basically quantitative research, there are different hierarchical way of the studies, starting from the case report, then the case series, cross-sectional studies, case control studies, cohort studies, randomized control studies, and then on the top we have the systematic reviews and the meta-analysis. Probably these are the few important research designs that one should have the basic knowledge, so that they can adopt the appropriate research designs for their studies. I'll briefly talk about these research designs. Suppose you want to do a prevalence study, you have to take up the cross-sectional studies. Like for example, suppose you want to know how many patients of diabetes mellitus visits your hospital. What is the percentage of the patients of hypertension in your hospital? How many of the patients are vaccinated in your hospital? Suppose you want to do this study. So you need a cross-sectional study. Incidence, suppose you want to see how many new cases will be added up, like how many new cases of COVID will be added up for the next 6 months. You need to do a cohort study. You can also do the correlational studies or causal association studies using the cohort and the case control. Suppose you want to see the prognostic studies, then you have to use the cohort studies. Or you want to see the nature of the disease, then you can take up the cohort studies. And especially the treatment effect, like how an intervention is going to affect over a particular condition, then you do a randomized controlled trials. So you can broadly classify these designs into the diagnostic studies, the descriptive studies, and the analytical studies. So some of the other studies which are commonly used, like we have the ecological studies, which is also called as the correlational studies. Suppose you want to correlate between a particular type of prakriti and a particular, like the hours of sleep. Or suppose you want to see what is the type of their diseases manifestation based on the prakriti. Suppose you want to see the correlation, or age and the blood pressure. So these are the correlation studies that you can do. And cross-sectional, case control, and the cohort studies, which need to be where you're trying to see the relationship between the exposure and the outcome. And of course, you have the intervention studies where you do the randomized control trials or field trials or the community-based trials. Cross-sectional is when an observational design is done and you try to do at a particular point of time. And the basic thing is it's also called as snapshot design, because at a given time, both exposure as well as disease status can be known at a single point. Suppose you want to know how many patients of diabetic patients are prevalent in a particular scenario, you can easily go for an observational study. Like for example, a socio-demographic patterning of a non-communicable disease risk factors in rural India, a cross-sectional studies, like how many of them are suffering with the various types of non-communicable disease, or like now at present, how many of them have been suffering with the complications of COVID-19, etc. So what are the pros and cons of the cross-sectional studies? Basically, though it is useful, it quantifies the prevalence rate, risk factors, the diagnostic test, and it has the advantages like it is cheap and simple, ethically safe. But there are some disadvantages, like it cannot establish the cause and effect relationship. There are recall bias, researcher bias, or there could be some confounders. The pros and cons of case control studies is like where the patient with certain case or outcome is present and you try to compare them with the controls without the outcome. and try to have the matching and then see whether a particular risk factors has caused a particular disease outcome. Advantages are you get a lot of information from the long case control studies, and suppose you are having any rare cases, it can be very well be used. The disadvantages are, again, the chances of recall or record bias and the selection of a control sometimes may be difficult in such scenario. The effectiveness of rotavirus vaccination against childhood diarrhea in El Salvador case control studies. Cohort studies where the data are, there are a group of people who have been exposed and those who are not exposed, and you follow them for a particular period of time. For example, those patients who are vaccinated, who are not vaccinated, and you follow for 6 months or 1 year, and you just see whether they again suffer with the COVID infections. These are like, it takes some long time. That's one of the disadvantages of this, but it is very important to establish the timing and directionality of the events. And especially we can standardize the studies. So these disadvantages could be because of it may take some longer time. Sometimes blinding may be difficult, and for rare cases you may need some large samples. So cohort study like the chronic kidney disease and risk of major cardiovascular diseases and non-vascular mortality, prospective population-based cohort studies. Comes to the randomized experimental studies, you have the randomized and the non-randomized. Randomized, what is called as randomized controlled trials. So randomized is done to eliminate the bias. Controlled is we have the control group, maybe active control or the passive control, and the trial is done because you have the experimental interventions. So here you have the proper hierarchical population for the intervention studies, where you have the reference population, then you have the two groups, intervention group and the study group. Sometimes you lose in the follow-ups, and then at the end you compare the outcomes. So the pros and cons of the RCTs, like especially the blinding is more likely, you can randomize them, but sometimes the disadvantages are that sometimes it could be ethically problematic, it could be expensive, etc. So you also can have the crossover trials, like where the individual becomes the self-controls. Like there are two groups. In both the groups, each one of them can be the one. Initially, a person will receive treatment A, and then after the washout period, again he'll receive the treatment B. And initially, sometimes people will start with treatment B. After washout period, they will start with treatment A. So for this, there are some reporting guidelines or statements which one should know as a student or as a person interested in research. This is very, very important. You can get all this information from the Equator Network website, where we have some standard guidelines for different types of study designs. Like suppose you want to publish a case report, you have the CARE guidelines. You want to publish the randomized control trials, you have the CONSORT guidelines. For diagnostic accuracy studies, the START guidelines. For observational studies like the cohort and the case control, you have the STROBE guidelines, PRISMA guidelines, and the MOOSE guidelines for meta-analysis of the observational studies. To summarize, the meta-analysis is a way of combining data from many different research studies. Meta-analysis is a statistical process that combines the findings from the individual studies. For example, the anxiety outcomes after physical activity intervention, meta-analysis findings. Systematic review is the summary of the clinical literature. A systematic review is a critical assessment and evaluation of all the research studies that address a particular clinical issues. And the basic difference between the systematic review and the meta-analysis is these reviews are again quantified and they are undergoing the analysis. Like analysis of analyzed data is called as meta-analysis. Like for example, complementary alternative medicine use among women with breast cancer, a systematic review. Randomized control trial, like a controlled clinical trial that randomly assigns participants to two or more groups. And there are various methods to randomize the study participants, like you have the lottery method, coin method. There are softwares, random number tables, etc., which can help you to randomize the study participants. Like example, meditation or exercise for preventing acute respiratory infection, a randomized controlled trial. The cohort study, which is a prospective observational study. So in this, a clinical research study in which people who are presently have a certain condition or receive a particular treatment are followed over the time and compared with another group of people who are not affected by the condition. Like for example, smokeless tobacco session in South Asian communities, a multicentric prospective cohort study. Case control study, researchers choose people with a particular result or the case and they interview the group or check their records to ascertain what different experiences they had. They compare the odds of having an experience with the outcome to the odds of having the experience without the outcome. Like for example, non-use of bicycle helmets and risk of fatal head injury, a proportional mortality case control studies. Cross-sectional studies, an observation of a defined population at a single point in time or time interval, exposure and outcome are determined simultaneously. Like for example, fasting might not be necessary before lipid screening, a nationally representative cross-sectional study. Case report and series, a report on a series of patients with an outcome of interest, and here no control group is involved. Like example, students mentoring students in a service learning clinical supervision experience, an educational case report. And there are also preclinical studies, especially for the safety and the toxical evaluation. You have the studies conducted using animal models, like the intranasal leptin reduces appetite and induces weight loss in rats with diet-induced obesity. So these are some of the examples and the research designs which one should know. And this is very, very important for any researcher. So this is just a brief orientation that we need to understand some of the newer research designs. And especially, I would like to emphasize on two important research designs, like the case control studies and the cohort studies and various types of qualitative research methods, which can be very, very effectively be used in the clinical studies related to Ayurveda. So I once again thank the organizers for these wonderful opportunities. And if there are any questions by the participants, I'm very happy to answer. Thank you. Thank you very much.
Thank you, Dr. Shetty, sir. And it's very good to get a vast knowledge regarding the research designs, mainly
Excellent.
qualitative and quantitative designs. Thank you once again, sir. And we'll be meeting again for
Excellent,
more
excellent, Dr.
enlightenment.
Suhas. Excellent.
Thank you.
And now I would like to call upon stage Dr. A.K. Jain. He is MD, PhD in Ayurveda, a senior diabetologist. He is expert in diabetes and his expertise in clinical research also. I would like to invite Dr. A.K. Jain, sir. Please highlight and enlighten us with your vast knowledge regarding research methodology. The stage is yours, sir.
Welcome, welcome, Dr. Jain. Welcome, Dr. Yunis Munshi also. Welcome. Yunis Munshi ji in charge of RRI Unani. Welcome.
Thank you. Thanks a lot.
Dr. Jain, sir, please unmute yourself.
Please allow me to share the screen.
Yes, sir.
Yeah, no?
Okay, sir, please start, sir.
Is it visible?
Yes, sir, it's visible.
Am I audible?
Yes, sir, you're audible.
So, dear friends, good afternoon to everybody. I am very much thankful to my dear friend, Dr. Pawan Sharma, who has taken this very painful job during this COVID period, and he continuously organized this webinar for the sake of Ayurvedic development. Now, the topic which is allotted me for this research methodology, I will take care of this Western methodology, then the second Ayurvedic methodology, I can recall this memory of all the research scholars for this Ayurvedic methodology, which is already forgotten by us. So now, first of all, we will take this Western methodology. What is the research methodology in Western history? And what is the research? The first of all, we have to define what is the research. Research is defining and redefining the problems. It's the formulating the hypothesis or suggested solution. It's the collecting, organizing, and evaluating data, marketing detection, and the reading conclusion, or at the last, carefully testing the conclusion to determine whether they are fit in formulating hypothesis or not. So this is the main aims of the research, which comprises. So the primary purpose of conducting research through the research methodology is to find answer of what the answer, why is research started, why we are starting our research, what are the reasons behind the formulation of the hypothesis, the hypothesis we have formed, what is the reason, and how can research problem be known, and what and from what sources data have been gathered, what specific method will be the best for this particular study. So the research method and methodology, there is the two terminology which we use in research methodology, research method and research methodology. The research method is referred to the methods which by the research person use for performing the research operation. And research method is to be understood when the scientifically research is to be done by the researchers. So all the method which help the researcher to carry out the research, and this involves the different step, scheme, and algorithm are termed as the research method. So what is the research technically, if we can consider, this include the experiment, test, and survey. On the contrary, the research methodology teach the various techniques which help in research and teaches how to perform test, experiment, survey, and critical studies. So the technique or the specific procedures which help the student to identify, choose, process, and analyze the information that we gathered in our research about the subject is called the research methodology. So what the research methodology allows us, what we can understand after doing this research in a particular subject. So how did the researcher reach to decide what data should be collected or what data should be avoided? This should be researchers should know how to collect it, the data collection method of the research, and how to analyze it. This is the most important part of the research methodology, which should be everyone researcher should be know. The role of methodology in solving the problems in the way of researcher, what the research methodology help us. It helps in deciding the best method to resolve the difficulties for the research, and research describes how efficient the method is in solving the problem. So the detailed study of the methodology of research teaches to select the best method, data, or information, scientific way, and teach the technique to solve the problem. So this is the research methodology. Now, Dr. Shetty had described the design of the research. Now I am giving the various type of the research. What type of the researches are in our practices or in the world where the scientists are busy in the different type of the research. So one by one, I am describing this is the applied research. What is the applied research? It refers to the scientific study and research that seek to solve practical problem. This is the applied research. So this is the use to find solution to everyday problems. It cure illness and develop innovative technology rather than to acquire knowledge for the knowledge sake. So this improve, actually the advantage of the applied research improve the agriculture crop production or treat or cure a specific disease. Now the basic research, the second number is the basic research or the fundamental or pure research, which is driven by the scientist curiosity or it interest in the scientific question. So this is for best example is how did the universe begin? What are the proton, neutron, electron compose? So the main motivation is to expand man's knowledge, not to create or invent something. The correlational research refer to the systematic investigation or the statistical study of the relationship among two or more variables. Now what's the example of this? The experimental is the group samples. I make one group listen to music and then compare to the blood pressure level. And again, the survey asks the people how they feel, how often they listen, and then compare of the both group. Now the next is the descriptive research, which refers to the research that provide an accurate portrayal of the characteristic of a particular individual's situation or group. So this descriptive research also known as the statistical research. In short, if we describe the descriptive research, it deals with the everything that can be counted and studied, which has an impact on the lives of the people which it deals with. Ethnographic research refer to the investigation of a culture. This is the ethnography which investigate a culture through an in-depth study of the member of the culture. So the purpose of the ethnographic research is to attempt to understand what is happening naturally in the setting and to interpret the data gathered to see what implication could be formed from the data. Now the next is the experimental research, which is an objective, systematic, controlled investigation for the purpose of predicting and controlling phenomena and examining probability and causality among the selected variables. So the advantage is the best established cause and the effect relationship, and this advantage is also that this is the artificiality, feasibility, and unethical. The exploratory research, which is a type of the research protected for a problem that has not been clearly defined. So the exploratory research helps determine the best research design, data, collection method, and selection of the subject. So this is the research is not typically generalized to the population at large. Grounded theory research. The grounded theory research is a research approach designed to discover what problem exists in a given social environment and how the person involved handle them. So this is the grounded theory research. It involves the formulation, testing, and reformulation of the proposition until a theory is developed. The next is the historical research, is the research involving analysis of the event that occurred in the remote or in the past. So the past, any events is happened in the past, that should be again be reinvestigated. It's called the historical research. The phenomenological research and inductive, descriptive research approach developed from the phenomenological philosophy. Its aim is describe an experience as it is actually lived by the person. So this is concerned with the study of experience from the perspective of the individual, taken for granted assumption and usual way of the person. So there's the broader way perspective. We can say the research can be classified into two groups, qualitative research and quantitative research. Dr. Shetty has already explained the qualitative and quantitative, so I am not going in detail. What is actually the research? Then the person is going for doing this MD or PhD in particular any subject of the research, but they have to think about the First is the research philosophy. What is the research philosophy? This is the positivist, realist, and interpretive. And then again to approach, what is the research approach? Deductive or inductive. And thereafter the research strategy. Then the student has to form the research strategy. Experiment, survey, or case study, or grounded theory, or ethnography, or action research. Then again to the time horizon, cross-sectional or the longitudinal, or thereafter the data collection, and data collection, the sampling, secondary data observation, interview, and questionnaires. So in nutshell, we can give a short idea of the research is the formulating the research problem. First of all, extensive literature survey should be done before going to the investigation. Development of a working hypothesis should be formed, and preparing the research design, as Dr. Shetty has told. The analysis of the data, thereafter, the determining the sample design, collection of the data, execution of the project, analysis of the data, and then hypothesis testing, generalization and interpretation of the data, and preparation of the research report. So this is the nutshell of any research work before going to do. So primary research method and the technique, the primary research is divided into qualitative or the quantitative. Then the Dr. Shetty had described already. The most important point, the most important things which we overlook is the pharmacovigilance. The pharmacovigilance is most important point to be considered in your any research. What is the pharmacovigilance? The science and the activities relating to the detection, evaluation, understanding, and prevention of the adverse drug reaction or any other drug related problem is called this pharmacovigilance. What are the major aims of pharmacovigilance? Is the early detection of unknown safety problem. It is concerned with the safety profile of the product, because safety is the must. Safety is the very much concern. The adverse reaction is very much concerned with the patient or as well as with the product. So detection of increase in frequency, identification of the risk factor, what are the risk factors are associated with this research, quantifying the risk, and preventing patient from being affected unnecessarily. So why the pharmacovigilance is important? Because the adverse drug reaction are the 4 to 6 largest cause of mortality in USA. This is the very, very important because the adverse drug reaction should be considered before going to into the research. So the why pharmacovigilance, the post-marketing topics, the unexpected adverse reaction should be collected from the post-marketing persons, interaction, dependency, long-term efficacy, if there is any resistance, what are the risk factors, what's the quality, and also the cost assessment which is concerned with the patient. So why this vigilance is important? The updating of the clinical safety profile, which is very much concerned with the product, identification of the new risk not recognized empirically, identification of the rare adverse reaction, updating of the benefit risk ratio and comparison with new option for the treatment, and also the updating information for the consumers and health professionals. So the adverse reaction is very much concerned with any clinical trial because the safety is very, very important part for being calculated. And now I will touch the research in Ayurveda. There are various challenges in research of Ayurvedic medicines. Various challenges. Those who are involved in the clinical research of Ayurvedic product or the experimental research of Ayurvedic product, really they are facing a very much challenges because there is no particular guidelines. Now the Ayush Mantralaya has given some guidelines. Previously it was not available. So what are the problems facing by the clinical investigator? problem is specific to the quality of the herbal drug, because the herbal drug are mixture of many constituents. Active principle is not hardly known. Selective analytical method may not yet exist. Different compound may not be available commercially. And the chemical variability of the plant material, because seasonal plants have the different type of the alkaloids and glycoside. Natural variants, chemo variety and chemo cultivars, influence of the harvest, drying and storage conditions. So these are the point which is the very much problematic for the herbal drugs research. So safety is the fundamental of the Ayurvedic product. And the clinical researcher, those who are facing the problems, what are the problems in clinical research? Because we have done a number of clinical research nationally and internationally on the GCP guideline. We know the problems with the Ayurvedic product, adulteration with other medicine and potent chemical substances, mistaken use of the wrong species, incorrect dose, and the misuse of the herbal drugs by healthcare providers and consumers, poor quality products, likelihood of the innate toxicity, and interaction with the other medicine. The most important part is the misconception that all natural are safe. This is the wrong conception, because all natural or herbal products are not safe. Method of analysis of herbal adverse reaction is observational, pre and post marketing surveillance, pharmacovigilance center reports and database, and literature report, and the research laboratories also give the adverse reaction. The method of the analysis of the herbal adverse reaction is observational. From the patient, consumer family, from the friends, we can come to know the pre-medication symptoms, physical and mental changes before starting the medication, and concomitant the prescribed medication which in the past the patient is taking. And in clinic and hospital, we also take the medical history, including this medication, botanicals, vital signs, symptoms, and also the laboratory test, the hematology, biochemistry, ECG, and other investigation which is related with the clinical trial. We have also take care of the safety and toxicity of the Ayurvedic product. We have to monitor this, because there is some sign and symptoms appears in the context of the adverse reaction. The acute sign of the discomfort, nausea, emesis, blood pressure and heart rate changes, dizziness, headache, diarrhea, diuresis, and laboratory abnormalities. And vital sign also monitored hourly in the clinical trial. So this is very much important. If any symptoms if any of one symptom appear with the clinical trial of any product, should be informed to the investigator immediately. Like conventional drug, adverse reaction in herbal medicine therapy also occurs. This reaction can be detected by the personal and medical practitioner observation, pre and post marketing surveillance, pharmacovigilance center report and database, literature report, and laboratory research. And some long or non-clinical testing of the herbal drug preparation with the long-term marketing experience. If there is the long-term marketing experience we have of any product, so the test is not required, because we have the sufficient experience of the product in human from the post-marketing surveillance at the single dose toxicity, repeated dose toxicity, immunotoxicity, local tolerance testing, pharmacological test, including safety pharmacology and pharmacokinetic studies. There is no test. The another thing is that this European The European Parliament and the Council have given the definition of adverse reaction. What the adverse reaction in GCP trial, they have clearly mentioned what is the adverse reaction, what is the severe adverse reaction, and how we can tackle it, how we can compile it, and how we can inform the principal investigator and the team or the concerned person. So the adverse reaction is the response to a medicinal product which is noxious and unintended and which occurs and that is normally used in men for the prophylaxis, diagnosis, or therapy. The serious adverse reaction, which should be considered as a result in death, is life-threatening or require inpatient hospitalization or prolongation of existing hospitalization, result in persistent or significant disability or incapacity or is congenital anomalies birth defect. So this serious adverse reaction should be noted, and some are the unexpected adverse reaction, that is nature and severity of the outcome of which is not consistent with the summary of product characteristics. So all the instruction has given that member states shall encourage doctor and healthcare professional to report the suspected adverse drug reaction to the competent authorities. And the member states shall establish a pharmacovigilance system, which is most important, collect information with particular reference to the adverse reaction and to evaluate such information scientifically, or take into this account information on misuse or abuse of medicinal products. So this should be informed to the member of the investigation team. The marketing authorization holder, they have mentioned that marketing authorization holder shall have permanently and continuously at its disposal an appropriately qualified person responsible for pharmacovigilance should be appointed, and that MAS shall be required to maintain detailed records of all the suspected adverse reaction, to record and to report adverse reaction. This is the most important because the adverse reaction is very, very much concerned with the product as well as with the patient. So in the nutshell, the research in Ayurveda, first to define the research problem, review the literature, widely formulate the hypothesis, design the research as already described, collection of data and analysis of data and interpret it, and the report is finally. So be prepared to be criticized having reacted too late or too early, and especially too big or too strong. So be prepared to be criticized. And this is the Ayurvedic, just I am touching this Ayurvedic the mention in the Ayurveda, what's the research methodology mentioned in Ayurveda? Praman. Praman can be considered actually because according to the Charakacharya, the things of the universe such as Sat or Asat can be investigated by these 4 Praman. And the same are helpful in Swasarachanam, Rog Paricha, Rogi Paricha, Dravya Paricha, and Chikitsa. What are these? Aptabdesh Praman, Prataksh Praman, Anuman Praman, and Yukti Praman. These 4 Praman should be used in our all the step in research for the Ayurvedic product according to this Ayurvedic as mentioned in our Charaka. So among all Pramanas, Aptabdesh gives existing knowledge of science, while Prataksh and Anuman gives the knowledge after the examination. So this is the Anuman. What the Anuman Praman says, and what the importance we have gathered from the Anuman. It is indirect knowledge which is based on the reasoning, or the inferences, and the inferences is based on the prior perception. Medha or grasping capacity or smriti can be assisted by the Anuman. So Charaka elaborates further with discrete example which refer to an inference of the present situation based on the available clue. So Anuman plays a major role in decoding the etiology of disease and in diagnosis. The next is Yukti. It means the intellectually planning on the basis of Prataksha and Anuman. That Yukti should be applied for this Ayurvedic research. The knowledge which see the things produced by the combination of multiple causative factors is known as yukti. So we can get the knowledge of three kalas and also three varga, dharma, earth and kama. And yukti proves to be important tool of the diagnosis and treatment when there is an involvement of the multiple dosas, presence of the confusing symptoms, and the availability of the few treatment options due to the contraindication in the disease and the constitution of the patient. So this is the doctor's side that we can give. This is the very confusing slide because the doctors should be the highly qualified with high ego, analytical mind, and business conscience should be there. And the doctor should act preclinical data, proof of efficacy, define the side effect, skeptical about quality, mode of action, presentation. So these should be the action of the doctors or highly qualified which is involved in the research. GCP trial, the last meeting, I think the GCP about the GCP trial is already discussed. Just I am giving you 2 lines that of clinical trials that provide assurance that the data and the reported results are credible, accurate, and that the right, integrity, and confidentially of the trial subjects are projected. So this clinical trial, now the GCP trial is applicable in each or every herbal product or any modern product because GCP trial is authenticated by this Western committee as well as this Ayush Mantralaya. So every product nowadays under the clinical trial will do on the GCP guideline. The clinical trial should be conducted in accordance with the ethical principles that have their origin in the Declaration of the Helsinki and that are consistent with the GCP and the applicable regulatory requirement. This regulatory requirement should be followed in doing this clinical trial on the basis of GCP. So just a few lines on the determining sample design, how we can collect this sample, step and define the universe. Because the sample design is the heart of the research work. What type of the sample we have to gather actually, what type of the sample design we have to make. So define the universe, sampling unit, source list or sampling frame, size of the sample, sampling method or technique, parameters of the interest and budgetary constraint and select the sample. So method of samples may be grouped into 2 broad classes according to the method of selection, namely random or probability sample or non-random or non-probability sampling. Random or probability sampling are the simple random sample, systematic sample, stratified random sample, multi-stage random sample, clustered sample, sequential sample, or the replicate or interpreting sample. So these are the major random samples we can use in collecting for the clinical trial, and major non-random or non-probability samples are purposive sample, quota sample, and convenience sample. So method of collection of data, this is the last point. We have to discuss the data because data is the very essential part of your research. This is the summary of your total research work. This is the conclusion of total research work. The data which you have gathered and analyze them and interpret with your basically theme. So while deciding about the method of data collection to be used for the study, the researcher should keep in mind 2 types, primary data or the secondary data. The primary data are the several methods of collecting this primary data is questionnaires, observations, interview, and schedule, whatever is made. And the secondary data means the data that is already available. It may be either published or unpublished. Published data are available in various publications of central, state, or local government. So public record and statistics, historical document and other sources of the published information should be considered for the collection of the secondary data. Sources of unpublished data are many and they include the diaries, letter, unpublished biographies and autobiographies. Data available with the research scholars and research workers, trade association, labor bureaus, and other public, private individuals and organizations. So we can gather this data from these are the association or bureaus and other firms. Then after collection the data, processing and analysis of data. After collecting of data, it has to be processed and analyzed. And the process of the analysis is the editing the data, coding, classification, and tabulation. This is the process of the analysis. After gathering all the data, we have to do this analysis for the further publications. And these are the types of analysis: descriptive analysis, correlation analysis, multivariate analysis. Under multivariates, we have the following analysis: multiple regression analysis, multiple discriminant analysis, multivariate analysis of variance, and canonical analysis. And last is the inferential analysis. And then we have to do the interpretation. Interpretation of the data means the task of or the drawing conclusion and of explaining their significance after a careful analysis of data what we have gathered after the clinical research. So these are the technique of the interpretation. The interpretation is required a great skill. The researcher should be in a great skill to interpret the data, and the part of the researcher, this is the very, very important, and it is an art that one learns through practice and experience. The interpretation is an art and it should be learned from the practice and experience. So this technique of interpretation often involves the following step. Researchers must give reasonable explanation. Extraneous information is collected during the study. It is advisable before embarking upon final interpretation to consult someone having insight into the study. Don't feel any ego or shy to take the consultation with the senior or who has done the research in the past on this particular subject. So researcher must accomplish the task of interpretation only after considering all relevant factors affecting the problem. And thereafter, the presentation of the report. After the data analysis, there is the presentation of report, preliminary page, title page, certificate of the supervisor, table of contents, preface, acknowledgement, listing of tables, listing of figure, and abbreviation. These are the method to follow before presentation of the report. Main text, introduction, objective, methodology, findings, limitation, and ultimately conclusion and the recommendation. What's your recommendation of the trial? Add matter, appendix, and biography, alphabetical order, books, report, journal references, name of author, title, place, publisher, volume with the page number, and in the last, the glossary. These are the methods to follow for preparing the paper or giving the report finally. Thank you very much. Thank you for patience hearing. I am thankful to all of you. Thank you. Thank you very much.
Thank you, Dr. Jain. It's a very nice presentation, and you have highlighted very specifically the process of data presentation and what are the GCP protocols followed in India and internationally. Thank you once again, sir. Now I would like to invite Lieutenant Colonel Dr. Devkulyan Maji for his presentation. Let me introduce him, Dr. Maji. He is MBBS, MS in Obs and Gynae, DNB. He did his post-graduation from the prestigious Armed Force Medical College in Pune. And he has worked in various capacities in different positions in Indian Armed Forces, and he have been traveling in India in different places for his postings. And at now he is posted in the Command Hospital in Kolkata, and his designation is also assistant professor. So he has a vast knowledge and experience in gynecology, obstetrics. As a clinician, as a researcher, he has also served in capacity in DRDO. So I would like to call upon Dr. Devkaran Maji to present his presentation. Dr. Maji, the dais is yours now. Thank you.
Good afternoon, esteemed Judge and Pilot. Am I audible?
Yes, you are audible, sir.
Thank you, Dr. Parikshit, for all kind word and kind interactions. And I like to thank especially Dr. Professor Pavan Sharma for giving me the opportunity to speak in this webinar. I just want to share my presentation first, and I'll keep on talking about research methodology. Actually, all my previous speaker and sir, they have already described all the highlighted portion which is required actually for all PG students. Now I will tell little bit about research methodology. How actually you will write. How actually you will write a paper that need to be published in a journal. That is also one of the important thing. Research is basically it's gathering information and it's finding the answer of unanswered questions. But as a clinician, as a doctor of all these Ayush or modern medicines, whatever it is, we are not actually in a kind of invention thing, not in kind of finding something which is discover something which is new. Sometimes data is also important, and data in today's world, data and information. These are the factors. These are the factors you need to step down on that, and then you need to improve upon. So data is important, but believe you me, when I was in my third MBBS or doing community medicine and all that, then sir told me, okay, you have to do some ICMR project. Then I was realizing I am a doctor, I am in applied science, I am supposed to see patient. But why sir is telling me you go in research, you do collect some data and you analyze and you interpret. So believe you me, that time this 3 Idiot movie was not there, but in my mind it could have been finding the same feeling. Sir, what sir is actually trying to tell me? Sir is trying to tell me in a way, as a doctor student, I never thought of. But believe you me, it is important. Now I am realizing why it is important. See, we have discovered fire way back, but still we are improving. There was a gas lamp, there was a fluorescent tube, CFL, LED. So in the process, this is research. And this we need to understand. Like in biomedical waste disposal, we all understand the scientists, actually your data scientists, those who are actual researchers doing higher maths, calculus, divide, dx and all that, they are doing actual core research. But like in biomedical waste disposal, we all know that segregation has to be done at user level. So in a doctor of clinical medicine, where we are seeing patient, actually data are with us. So we need to understand, we need to have some basic knowledge about research and research methodology. Without research methodology, I am telling you, I can give you two examples. One example, in 2001 or 2001 to 2006, you all must be agree with me, the phone means Nokia. That bada kala dabba wala phone and Nokia. That company was so reputed, so worldwide growth. They just not interested in doing R&D because they think this is the end. We are having huge revenue and we will sustain. That time Samsung come and Samsung just doing R&D. They are going on Android display and all that. Now see that today's market. Have you ever seen any Nokia phone existing in this particular today's mobile market? So that is the importance of research. And as a clinician, we are actually inside the plethora of data. We just need to collect it and we need to represent it. That is the most important. You may take help from all other scientists who are actually doing core scientists, but data collection and understanding research is important. Why? We all know Sushruta. Sushruta is father of our Indian medicine. Even in modern medicines, we actually tell that Sushruta is the father of modern plastic surgery. Now, in Ayurveda, Siddha, Unani, these are all Indian traditional medicinal systems, which are actually promoted by all the king and kingdom, all the king and all the, even in Asoka era, Ayurveda flourishes. Not surgery, it is a medicine, which is a charak, because ahimsa, I want ahimsa. I don't want to do surgery, because otherwise prior to that, there was a time, even in Ramayana and Mahabharata, we all have read, the punishment is cut their nose and throat. So that is the actual indications key we need to improve upon. We have disfigured someone. In Britishers and all that, they used to cut, they used to kill. But in our system, our kingdom system, our ruling system, if somebody is doing something wrong, okay, cut his nose, cut his ear. Now that person is surviving, and that person is having lot of comorbidities later on. So there comes the Sushruta who use all those plastic surgery. The R&D has been done by some allopathic medicines, and they regarded Sushruta as a father of plastic surgery. But in traditional Ayurvedic medicine, because of Asoka and everything they have come, we fail to recognize. We have not probably done that kind of R&D to recognize Sushruta. But still, he is the father of modern Indian surgery. There was a time, you all must be agree, that all instruments used by Sushruta is even used nowadays today. So this is the importance of research, which I have talked about. Now I am coming to the research methodology. Methodology is how you will conduct the research. So what is research? Research is for the purpose of acquiring knowledge, and it is a systematic inquiry to describe certain things. Now methodology is what? Methodology is how you will do it. So significance of research with some, what is it? There are certain fundamental points. How you will build your knowledge, how you will nourish and open up your mind, different understanding issue, and things, how you go about it. So the objectives of my presentations will be, all researchers, all clinicians who are actually in the applied science, adequate level of information, they should have access to the adequate level of information, conduct, analyze, interpret, and write up your research. Assist you in this presentation will assist you in deciding well in advance what you may require. Contact right person in right way at right time with at right place. These are also important. Now, for many of us, for doctor who was actually practicing clinical medicines, I am repeating the same thing. It is an additional burden. Nobody is actually interested in to do research in a proper way. But there are other group also who actually want to publish and perish. This group is also coming up in a very big way. So I'll tell you 7-8 steps how to formulate a research methodology. See, whenever we are seeing patient, according to zone, if I belong to Kolkata, somebody belongs to Kerala, somebody belongs to Jammu Kashmir, somebody belongs to London, there is a variability of age group, variability of the disease, everything is variability is there. So now if I want to see how many portion of the population is suffering in worldwide, actually how to do, how a research has to be done, that all world population is to be collected, and all world population has to be collected upon, and they will be scrutinized, they will be questioned whether they are having disease or not. They should be examined and screened about. But practically this is not possible because it is very time consuming. It cannot be done in a stipulated time period, or that adequate economic funding won't be there. So variability of everything according to zone is there. So now I want to conduct a research. So what should I keep in mind? That I am doing something in a particular area of Kolkata. Now Kolkata people have certain proportion of disease which is not there in the people of London. They might have some disease which is more in amoebiasis. For UK people developed countries, they may not have amoebiasis as common as we find in Kolkata. But they might have some lifestyle disorder which is more common among them, which is not there among the people of Kolkata. So whenever I am doing this question, it should put on your mind that my research should not lose the external validity, that it should be generalizable. Whatever result I am getting, that should be generalizable. And whenever I am choosing the sample size, the population under study or the study population or sample size, my sample should out of sampling error. There should be precision in detecting sample size. I will explain every point after I just point out what are the points you should keep in mind. Then I'll keep one or two odd points explain about. Now comes the internal validity bias. So whenever you are conducting a study, because the amount of study data, everything has been explained by my all previous speakers. So now, what is the internal validity? How I will represent the data? How I will tell what is to be done? What is the measurement I will do? Now, I have collected all the data. Like, is there any indirect association or confounding factor? Like I am telling you, I want to see a research has been organized in an industrial area. There is a lot of population, lot of pollution. So I just wanted to see the pollution and association of lung cancer. So now, if you see that lung cancer is happening because of pollution, so I will get a data. Now, there will be certain proportion of people who smoke. Now, smoking has an association with the lung cancer. Now, if you don't think of this prior to your conducting the study, that is basically methodology. What will you find? You will find a confounding result. So there will be, you have to think of all this so that you can minimize your confounding factor. So confounding is one kind of bias only. You have to eliminate that. Now, what statistical and clinical significance you have? How will you represent your data? How will you tell that this is my data and what are the test you will do? What are the measurement you will tell? Whether you will tell mean, median or mode? How will you go about? This is very important, and you have to point out all this prior to start your study so that a robust methodology can give you a robust result. Then correct your research design, because you are thinking something, you have aim and objective is something, but you are giving a data or result something different. So if you are finding something different prior to starting your study, you formulate, analyze, and correct your research design you require. Whether you are going for prospective study, retrospective study, bidirectional study, or you want to do some clinical trial. But in this matter, I just like to tell you one thing: start with case reporting, then do some retrospective study where you will actually want to know how to play with the data. Then you will go for some prospective study, then case control study, then RCT. Initially, don't jump into the case control study doing RCT and case control, because for a beginners, it will be little bit difficult. Now, your research might yield something which is not answered in your research. So this will open a new direction for your further research. That is also very important. So because of something like, if I am doing post-graduation, I may not be interested, because this is a mandatory requirement. I have to do a dissertation and I have to submit it at a university to get my MD. But if I am doing something and I have something left unanswered, it will always keep in my mind, ki, okay, this question is answered, and I may choose a further study to go about it in the next run once I completed my MD. But if I don't complete it, then obviously my MD will be under question. So now this is the 7-8 point you always keep in mind trying to formulate in your methodology. Like all the way, how will you go about prospective study, whether your data is ordinally, these have all been explained by all my students. So I'll not go into all that. Now I'll go you on simple example. How will you describe this? Now, a first year MBBS student or first year BMS student for that matter, in a first year there are 25 students. These are the age group. I have written all the age group in a chronological order. So now I am doing a retrospective study. For example, 2019 May, 25 student has been admitted in our college. The age group is this. Now I want to just simply represent the data in your in an open forum, in a research perspective. How will you want to go about it? So this is the frequency distribution table or histogram you can do. Now this green line is your normal distribution curve. So now we can go, it is okay, the mean of the data is this, standard deviation is this, n value is this, we can go about that. Sorry. Now, if you analyze all this descriptive data, you will find that mean age of the statistical population, the age group is 22. We get the 95 continuous interval. In a software, you will get everything. You get median value, you get standard deviation, range, interquartile range. Now, where to use it? Where you will use mean and where you will use median? That is most important. Because you know everything, but whether this data requires mean or this data requires median, that is important. That you need to write in a proper way. So this is a box plot where you can see median value is 22 and I am seeing the range, and this is a one thing. So this is one data I am telling you. So this is one kind way of representing something. Now I will come little bit later. What is the significance? How to go about it? That I will tell later on my later part of my study. Whether I will go for mean or median, that test of normality, or I will tell later on. That I will come later. So in a nutshell, prior to formulating one research methodology, you need to ask yourself first this question, and then you will formulate your research methodology in this 8 questions chronologically. So once you do that, you will definitely able to make a right research methodology. So what are the steps of research methodology? First is ask yourself what is the correct research question. That my previous speaker already have told. Then what is the correct research question? It will actually give you the research in a conduct in a relevance issue, whatever it is there, it will able to do that. Now then another is selection bias. Selection of correct study design, that is also important. Why? Now correct study design has to be there, whether you are going to do a retrospective study, prospective study, RCT, or case report. So that's every study design has its own statistical stage and own way of representation. Like one of my previous speakers are already showed that you will go for consort guidelines or STROP guidelines. That you need to understand it, and that you need to think of prior to start your study, not after study. Now, adequate sample size. That is also very important. How sample size important? I will give you a simple example. Because I have already told, it will reduce the chance phenomenon. It will reduce the reduce the random sampling error and improve precision. So, how important is sample size? Like in a Kumbh Mela or whatever it is, a person is tossing a coin. So I have told 4 of the guys, ki, okay, I want to see how many percentage is coming as a head what is the proportion of head that particular is doing, and what is the proportion of tail. Now 4 students I have chosen, and 4 students went to that particular person, and they are standing and counting. One student, what they did, they just see one sample. Okay, one is head, so 100% head, no percentage of tail. Some next student, they just calculate 10 sample, and they see, okay, 8 are head, 2 are tail. So 80% chances that whenever you toss a coin, 80% chance you will have head. Now some choose 100 sample. Now whatever the finding, he find that 65 have head and 35 is tail. So you will tell, sir, in my finding, my research, 35%, everyone is right. Everyone is right in their finding. They have done it methodically. Now somebody has counted 1,000 times that person has counted that tossing of a coin, and her finding is 53% head and 47% tail. Now there is a problem. Who is actually, everyone is right on there. I cannot say you are wrong, you are wrong, you are wrong. So who is actually right? As a statistician, as a the person who is actually in the core research, how will you say, okay, that this person is probably methodologically wrong, this person is methodologically right? Here comes the importance of sample size. Now there are certain formula to calculate it. You can calculate sample size if you know the proportion of the disease. Proportion of the disease, like for example, I want to see that 10% population is diabetic in Calcutta. So I want to conduct a study that I want to see that among the study population of Khidirpur or among the study population around Command Hospital, I want to see the incidence of diabetes. So in that case, from that 10%, I will just simply calculate the formula n equal to z square by p and q by d square. You all must be knowing, or if you can actually read that and you will able to calculate the sample size. Now, if you know this is a proportion, you know there are certain instance where proportion is not known. So such a large population in a proportion is not known. Now, like for that example, I just wanted to tell you that I want to see the mean, what is the general range of blood sugar among the study population among Kolkata. Now I am, as you in a study, in a reported study, it is written that normal range of blood sugar is 80 to 90 or 100 for that matter, and standard deviation is 5. So if you know the standard deviation, if you know the then you can select your power and you can select your confidence interval, and then you can calculate your sample size. So this is also one of the important thing you must do prior to going into the study. So now I'll come. So why the sample size is important? Because you have to select the sample size by proper method, so that whatever you are doing, you are doing a research, you are investing some time, you are investing some economy, there is some financial connotations, there is some most important thing, your brain, you have to invest lot of physical effort, your brain is working, and sometimes it is a requirement for your education also. So proper sample size selection. You are doing something, you have worked on something and you did not get any result because methodologically you were wrong, then it will be little bit difficult. Now comes the correct tool. Like in a software, when you analyze the data, you will get mean, mode, median, everything you will get. Even you will get odd ratio, relative race, likelihood ratio, everything you will get. But which one you need to tell? Which one you need to represent? What is the correct tool? How will you compare between these two mean? Whether you will go for certain t-test or Mann-Whitney or parametric. These are also very important. And prior to studying your study, you should have a roadmap. That is most important. So now I will give you another example. Second year student, 25 student. This is the age group. So now this histogram and that histogram, there is a difference. Here you can see the normal distribution curve, but this data is not normally distributed. From apparent visibility, it is not normally distributed. But how will you calculate it? So you need to go with certain test. So here also you will get mean, mode, median. That median is 20, mean is 20.80, standard deviation is this, range is this. You will get the positive skewed value and kurtosis. So that means data is right-sided skewed. Even box plot, you can see that this is 25 percentile and this is 75 percentile. There is no outlier. So now comes the whether this data is normally distributed or not. So there are two tests you can do. One is case test, and case test shows the significance. That means there is no significant difference. There is a significant difference among the distribution of the data. So that means this data is not normally distributed. And if you see the previous study, previous sample, previous example, I'll just go back. So this is a chart. This is normally distributed data. And if you see that case test and Sapir-Whorf test, then you will find there is a difference among the data is not significant. As the p-value, if you take the 5% alpha error or p-value is 0.05 greater than 0.05. So this data is normally distributed, and in normal distributed data, it is better to represent the data in a mean and standard deviation, mean and 95% confidence interval and standard deviation. But if the data is not normally distributed, mean is not that important, then in that case, what will be important? The important will be median. So in that data, it is better way of representation will be the median age of our student. This student group is, for this data I will represent 20. 20 is the median age with interquartile range is 3. So that means 17 to 23 is the first quartile to, so this is the interquartile range will be 3. So from that you will do in this way, where in a data which is normally distributed, there you will write the mean data will be, for example, if the mean is previous study. So in this case, the mean is 22. This will be the, even you see that mean and median both are same. So mean is 22 and with standard deviation of 2.04 and with 95 to continuity. So this will be the way of representing your data, and you must follow this methodology to write a paper. Now next, confounding effect. I have already told that you need to think prior to conduct your study. Once you have done it and you have analyzed your data, then somebody tell, okay, you have not taken consideration of smoking as a confounding factor, because smoker will have the high proportion of lung cancer than that of non-smoker. So if you say because of this pollution, because of this factory, the people are having lung cancer more, but factory worker they smoke. So if you don't take into the consideration, then your result will be not be externally valid and it will not be accepted in a scientific form. Then come the appropriate statistical stage. This is again a very vast topic. You have to find the significance, you have to compare the means, and there are a lot of tests by which you can do, like t-test, chi-square test, man-Whitney, Kruskal-Wallis. These are all parametric, non-parametric. You all must be aware, and you all must take consideration prior. This is most important. Once you are going for a study, your every methodology roadmap has to be ready prior to recruiting one single case in your research paper. And whatever the finding you got, don't try to make anything big. Okay, I have to make it p-value less than 5. I want to make it significant. No. Ideal way of doing research is interpret with data, and whatever finding you got, you just represent that data according to the valid statistical set, and whatever result you get, you must do that. So in an art cell research, the more you go get into the research, more you will learn. It is a self learning. You start how to get into the research. First you start with case report, then you do some retrospective or prospective study, observational study. Then if you have, then you will grow interest, you will go for case control and all that. Then you can do cohort study at cities and all that, because these are all validation, and international research body, they have their stringent validation system, because you cannot do research on paper. You have to do something on ground, and then you represent. That is the best way of representing or doing clinical research. So now comes the what are the purpose and everything. This is a research design, where all you will, how you will go about. So don't get afraid by all this technical jargon. Start your journey with case report or retrospective study. Ask your question, these 8 points prior to conduct your study. Formulate a robust methodology, and believe you me, you all will be able to do such a research, and which will be accepted by all the good journal which are there in the medical science. Thank you so much, sir, and I have finished my all presentations. Now I will anyone having any questions, I'll be happy to answer.
Thank you, doctor. Excellent, nice presentation. Excellent. So a question answer session will be just after Dr. Rajiv's lecture, that we will arrange after Dr. Rajiv's lecture. So Dr. Parikshit, excellent, doctor.
Thank you so much, sir. Jai Hind.
Jai Hind, Jai Hind.
Now I would like to call upon our next speaker, Dr. Rajiv Purre. He is MD Ayurveda and from HND University Srinagar, BMS, BSc, MBA, and PhD. Currently, he is working as an associate professor in Department of Rasashastra and Vaishajya Kalpana in Uttarakhand Ayurveda University. He has a vast academic experience and research experience and contribution, and the list is very long. And he is an expert in Ayush, Government of India, in different capacities and parameters. He has served in many capacities and also represented India in different foreign locations. So he has also been awarded many fellowships, and his area of specialization is Rasashastra and Vaishajya Kalpana and Ayurvedic pharmaceuticals. So I would like to call upon Dr. Kulla to present his, give the presentation and enlighten us about and share us about his knowledge. And I would like to call upon Mr. Dr. Purre, and please enlighten us. The stage is yours, sir. डॉक्टर
सर,
प्लीज
आप सभी को बहुत बहुत धन्यवाद। भगवान धन्वंतरी हमारे गुरुदेव के चरणों में वंदन करने के बाद बात रखूंगा। ओम धन्वंतरे नमः। अखंड मंडलावतारम। वॉइस आ रही है सर?
यस, क्लियर आ रही।
ओके भगवान
Your presentation is not visible. Is there any problem, doctor? Your presentation is visible.
ओके, वॉइस आ रही है सर?
हाँ जी.
Yes, sir, it's coming.
हेलो,
Yes, sir, your voice is audible, sir.
Your voice is clear and loud, doctor. Your screen is visible.
जी,
Can
जी.
you proceed?
तो आज जो है आयुर्वेद में, जी जी, आज आयुर्वेद में
आपके यहाँ नेट
थोड़ा
कमजोर
सर
है
यहाँ
लगता
इंटरनेट
है.
का प्रॉब्लम है, तो डिस्टर्बेंस हो रहा है. अगर कोई
हाँ
तो
तो
सर
आप
मैं ये
वीडियो
कहना
बंद कर
चाहता हूँ
देंगे
कि
तो
आयुर्वेद
हो
में
जाएगा.
रिसर्च की आज के परिप्रेक्ष्य में बहुत ज्यादा आवश्यकता है, क्योंकि हम लोग रिसोर्स में, समृद्धि में, ज्ञान में, विज्ञान में बहुत आगे हैं. हाँ, यस सर, यस सर. ओके ओके ओके. सर
Please tell me what is the problem.
ज्वाइन किया,
Yes, yes, slide is moving.
डिस्टर्बेंस. तो मेरा आज विषय है रिसर्च मेथड. बहुत से हमारे सम्मानित स्पीकर हैं, जैसे कि जी डॉक्टर मांझी जी, डॉक्टर सुहास जी, सभी ने लगभग सारे एरिया को कवर कर. डॉक्टर साहब हमें को तो मेरे साथ सारी बातें हो गई हैं. तो मेरा ये कहना है कि आयुर्वेद में आप सारी दुनिया में स्थापित करने के लिए जो 5 एरिया है जिन पे हमें फोकस करना चाहिए, उसमें पहला फोकस है क्वालिटी एजुकेशन. क्वालिटी
राजीव सर, आपका वॉइस कुछ आ नहीं रहा है अभी भी, तो आई थिंक नेटवर्क प्रॉब्लम चल रहा है. तो आप Hello, Rajiv sir, are you there?
Rajiv ji, please share your presentation again.
Rajiv sir, please share your presentation, sir.
I think in Uttarakhand net problem is there.
Yes, yes.
He is from Haridwar, very nice city.
Pawan
Welcome,
sir,
Dr.
can we
Kamil.
do,
Welcome, Dr. Kamil.
Pawan sir, can we do one thing? We can ask Ms. Dr. Rajiv to send the presentation and you can share from your
Thank you, sir. Thank you.
computer.
Rajiv ji, can you send me your presentation on my WhatsApp? That's why I am asking from every speaker presentation to avoid this technical problem.
Dr. Rajiv, are you there, sir? Dr. Rajiv is not audible also at this moment, sir. Yeah, Dr. Rajiv is there. Sir, can you share your presentation, please? With Dr. Pawan Sharma.
हेलो सर, आप लोग सुन पा रहे हैं सर?
अभी सुना
हाँ,
सुनाने दे
clear
रहा है,
सुन
सर।
पा रहे. Yes, yes,
अभी
yes.
सुना दे. Yes, sir. Okay, sir, please start your presentation then.
हेलो,
Yes, sir, you're audible, sir. Yes, sir, you're audible. Please start your presentation.
Hariom, Dr. Rajiv Pur. Rajiv ji has joined again. I hope there will not be any problem. राजीव जी, क्या मेरी आवाज आप तक जा रही है? राजीव जी, डॉक्टर राजीव,
सर हेलो।
हाँ, आप बिना प्रेजेंटेशन के भी बोल सकते हैं। 8-10 मिनट गुजर चुके हैं, आप बिना प्रेजेंटेशन के भी बोल सकते हैं।
जी सर। सर प्रेजेंटिंग का होस्ट को परमिट कर दीजिए तो मैं प्रेजेंट कर पाऊँ स्क्रीन।
यस प्लीज
हाँ
प्रोसीड।
जी थैंक यू सर, थैंक यू। सॉरी फॉर डिस्टरबेंस। तो मैं ये कह रहा था सर कि जितने भी जो की एरिया है जहां हमें फोकस करना चाहिए वो क्वालिटी रिसर्च, क्वालिटी एजुकेशन, क्वालिटी ड्रग, कंजर्वेशन ऑफ मेडिसिन प्लांट एंड अवेयरनेस ऑफ आयुष सिस्टम। तो आज ये क्वालिटी की बात कर रहे हैं। एजुकेशन तो सदियों से चल रही है, हमारे पास है। रिसर्च की बात कर रहे हैं, रिसर्च अभी नहीं हो रही है। और रिसर्च में कैसी क्वालिटी होनी चाहिए उसके लिए अभी बात करेंगे। फिर क्वालिटी ड्रग्स, मतलब सारी दुनिया में एक जैसी क्वालिटी हो दवाई के लिए, चाहे वो छोटे से गांव में ले या मेट्रो सिटी में ले या अमेरिका में ले या लंदन में ले, कहीं भी ले, दवाई की एक अपनी विश्वसनीयता, ऑथेंटिसिटी और उसकी पहचान और उसका स्टैंडर्ड होना चाहिए। तो ये सब लाने के लिए, ये स्टैंडर्ड लाने के लिए जो हमें मदद मिलने वाली है वो रिसर्च से मिलने वाली है, क्योंकि R&D ही किसी भी देश के, किसी भी संस्था के, किसी भी सिस्टम के विकास का एकमात्र कारण होता है। रिसर्च, रिसर्च दो शब्दों से मिलकर बना है। हमसे पहले हमारे सम्मानित डॉक्टर जैन साहब, शेट्टी सर, सुहास सर, सभी ने जो है, डॉक्टर मांझी जी, लगभग सारे एस्पेक्ट को कवर कर दिया है। लेकिन रिसर्च के जो बेसिक कुछ चीजें हैं जो आयुर्वेदिक एस्पेक्ट हैं उन पे मैं चर्चा करूंगा। रिसर्च शब्द जो है, रिसर्च फ्रेंच शब्द से बना है। इसके दो मतलब होते हैं, री और सर्च, जिसको बार बार खोजना, रिपीटेड जो है सर्चिंग, अगेन एंड अगेन खोजना। तो क्वेश्चन ये आता है कि एक चीज को, अगर कोई चीज है तो क्यों खोजना, बार बार क्यों खोजना? तो खोजना इसलिए क्योंकि सत्य की खोज करनी है, और इस सत्य को जो है मानने के लिए एक नहीं, हमें कई सारे एविडेंस चाहिए, जिससे कि आजकल एविडेंस बेस्ड मेडिसिन जो एक टर्म चल पड़ा है सारी दुनिया में। तो सत्य की खोज चाहिए, लेकिन सत्य तो पहले से एग्जिस्ट है। उस सत्य की खोज के लिए हमको सबूत चाहिए, एविडेंस चाहिए, प्रमाण चाहिए। और वो कैसे चाहिए? किसी एक व्यक्ति ने कह दिया, डॉक्टर शेट्टी जी ने कह दिया है, डॉक्टर जैन साहब ने कह दिया, डॉक्टर पवन शर्मा जी ने कह दिया, तो उतने से काम नहीं चलेगा। हमें साइंटिफिक एक एविडेंसेस चाहिए, एक कलेक्शन चाहिए। तो इसीलिए इसमें क्या है, किसी भी चीज में अगर एक बार दवाई से कुछ खोज हो जाती तो काम बंद हो जाता। तो रिसर्च क्या है, कंटिन्यूअस प्रोसेस है। इसकी इटिमोलॉजी पे बात करते हैं कि ये जो है टू गो अबाउट सीकिंग। इसी को चरक ने जो है ज्ञान के पैरामीटर
Sir, you are not out. Dr. Ghazi, if you are not audible.
तो महर्षि, हाँ, महर्षि चरक ने जो है अपने श्लोक के द्वारा बताया, विद्या वितर्क विज्ञानम स्मृति तत्परता क्रिया षड्गुणम एते ना असाध्यम ना अति वर्तते। अर्थात एक साइंटिफिक सीरीज बता दी उन्होंने। और इतना बेहतरीन तरीके से बताया कि आप सारी रिसर्च मेथोलॉजी को ले लें तो इसी में आप कंक्लूड कर देंगे। तो माने फैक्ट, जो विद्या अलग है, वितर्क अलग है, रीजनिंग जिसमें की सारी मेथोलॉजी आ जाती है आपकी, और विज्ञानम, विज्ञानम जो है विभिन्न प्रकार की क्रियाओं के द्वारा जो एक साइंटिफिक फैक्ट निकल के आते हैं उनके बारे में चरक ने बताया। और स्मृति, उसके आधार पर कुछ डाटा कलेक्शन करना, कुछ जो है ऑब्जर्वेशन इकट्ठे करना। तदुपरांत उन्होंने क्या बताया, स्मृति तत्परता, रिपीटेड ऑब्जर्वेशन, बार बार जो है क्लिनिकल ट्रायल्स करना है, दवाइयों की खोज करना है, एफएनडी करना है, क्लिनिकल ट्रायल करना है, दवाइयों की जांच करना। फिर क्रिया, उसके अप्लाइड एस्पेक्ट पे बात करना। तो चरक में सब कुछ दिया हुआ है, लेकिन बात यह है कि जो ज्ञान और विज्ञान है, तो अभी हम लोग सिर्फ संस्कृत में उलझे हुए हैं। आज जो है आयुर्वेद इतना समृद्ध शास्त्र है, इतना समृद्ध विज्ञान है कि हमारे पास सब कुछ है, शब्द है, लेकिन उनको पाश्चात्य सभ्यता के लोगों को समझाने के लिए हमारे पास सबूत नहीं है। सबूत के लिए, इकट्ठा करने के लिए ही रिसर्च की जरूरत है। इतना समृद्ध विज्ञान है कि आप अगर अपनी जो है चिकित्सा पद्धति में उसको प्रयोग कर लेते हैं तो किसी को इब्बट की जरूरत नहीं, कोई डाउट की जरूरत नहीं। हर जगह आपको जो है या भी क्रिया भी अभी जायन्ते शरीर धातु समा चाह चिकित्सा विकारणम कर्म तद विज्ञान मतम, माने दुनिया को एक ऐसी चिकित्सा दी जिससे धातुओं के साम होते हैं, इक्विलिब्रियम होते हैं, और साथ चिकित्सा विकारणम, ऐसी चिकित्सा करते हैं जिसके द्वारा कोई विकार उत्पन्न नहीं होते हैं। और इसी चिकित्सा की तरफ सारी दुनिया देख रही है, लेकिन हम क्यों सफल नहीं हो पा रहे हैं? क्योंकि हमारे पास रिसर्च के एविडेंसेस नहीं है, और एविडेंसेस को कलेक्ट करने की जो विद्या है वो R&D कहलाती है, रिसर्च कहलाती है। तो रिसर्च में जो है वैज्ञानिक पद्धतियों का अध्ययन करते हैं, और विज्ञान संगठित सुविशिष्ट ज्ञानम विज्ञानम, एक सिस्टमेटिक नॉलेज जिसमें कि 5 बातें रहती हैं, जनरलिसिटी, एक्यूरेसी एंड सिस्टम। इनके द्वारा जो है हम 4 प्रकार की साइंटिफिक टेक्निक के सहायता से हम ऑब्जर्वेशन को इकट्ठा करते हैं। देयर आर द 4 वाइटल फैक्टर फॉर द साइंटिफिक पैरामीटर। वन इज ऑब्जर्वेशन, हाइपोथेसिस, एक्सपेरिमेंट एंड कंक्लूजन। तो जो भी रिसर्च है इसकी डेफिनेशन पे न जाके, क्योंकि ऑलरेडी टाइम हमारा काफी हो गया है, ये WHO ने डेफिनेशन दी, रिसर्च इज द क्वेस्ट फॉर नॉलेज थ्रू द डिलिजेंट साइंस एंड इन्वेस्टिगेशन और एक्सपेरिमेंटल एम्ड एट डिस्कवरी एंड इंटर रिलेशनशिप ऑफ द न्यू नॉलेज। अर्थात इन्होंने कहा कि ये ज्ञान की प्यास है, और जो है रिसर्च भी इसी तरह 2 प्रकार की होती है, अभी आगे मैं आपको बताऊंगा। तो इस ज्ञान की पिपासा को पूरा करने के लिए हम रिसर्च का प्रयोग करते हैं। इसके द्वारा हम खोजते हैं, किसी चीज का इन्वेस्टिगेशन करते हैं, डिस्कवरी करते हैं, एक्सपेरिमेंट करते हैं, और कई बार जो खोजी हुई चीज है उनका इंटर रिलेशनशिप भी करना, जो सेकेंडरी जो खोज है, सेकेंडरी रिसर्च जिसकी बात अभी मैं बताऊंगा। तो ये WHO। इसी तरह अगर हम शॉर्ट में कंक्लूड करना चाहें, रिसर्च इज अ सिस्टमेटिक कलेक्शन, एनालिसिस, इंटरप्रिटेशन ऑफ द डाटा टू आंसर ए सर्टेन क्वेश्चन और सॉल्व ए प्रॉब्लम। तो इससे हमें 2 बातें समझ में आई कि जो रिसर्च है, रिसर्च के 2 उद्देश्य होते हैं। पहला उद्देश्य होता है ज्ञान की वृद्धि करना, फॉर द रिसर्च डन फॉर द सेक ऑफ नॉलेज, फॉर द सेक ऑफ इंक्रीजिंग नॉलेज। जिसके पास ज्ञान नहीं है वो ज्ञान की खोज के लिए करेगा, जिसके पास है वो बढ़ाने के लिए खोज करेगा, और जिसके पास बढ़ाने वाला भी ज्ञान है वो बांटने के लिए खोज करेगा। तो ये रिसर्च का पर्पज है। इसी को कहते हैं, दिस इज द फंडामेंटल रिसर्च। जब भी ज्ञान के लिए, जब भी ज्ञान को बढ़ाने के लिए, ज्ञान को बांटने के लिए रिसर्च करी जाती है, वो फंडामेंटल रिसर्च कहलाती है। जब भी संकट मोचन, प्रॉब्लम को सूट करने के लिए, प्रॉब्लम को खत्म करने के लिए, समस्या के समाधान के लिए जो रिसर्च करी जाती है वो व्यवहारिक रिसर्च कहलाती है, उसको अप्लाइड रिसर्च कहते हैं। और जो रिसर्च है, दो तरह की होती है, क्वालिटेटिव रिसर्च, क्वांटिटेटिव रिसर्च। जहां पे हम अमूर्त चीजों को बताते हैं, जहां पे हम किसी विश्लेषण के द्वारा, किसी के जैसे ब्लड है, ब्लड का क्या प्रकार है, आपकी जो है एथनिक ग्रुप है, कि आप एशियन हैं तो आप कितने एशियन हैं, इस डिग्री में नहीं बताते ना, आप भारतीय हैं या नहीं है, आप जो है एशियन हैं या नहीं है। लेकिन आपका ब्लड ग्रुप ए है या ओ है या डी है, जो भी है आपका बी ए बी है, लेकिन ये नहीं बताते कि मेरा ग्रुप ए बी बहुत बड़ा है, बहुत छोटा है, या इस अंशों में है। तो जहाँ पे कोई ग्रेडिंग ना हो, जहाँ पे खाली विश्लेषण के द्वारा, किसी घटना के द्वारा, किसी बात के द्वारा किसी का वर्णन किया जाता है, तो वो क्वालिटेटिव कहलाते हैं। और जहाँ पे किसी माप के द्वारा, किसी संख्या के द्वारा, किसी प्रमाण के द्वारा किसी विषय को प्रस्तुत किया जाता है, किसी डाटा को प्रस्तुत किया जाता है, वो हमारा क्वालिटेटिव डाटा कहलाता है। इसी तरह हमारा रिसर्च जो है हमारे दो तरह से और बांट सकते हैं, लेवल ऑफ इन्वेस्टिगेशन। तो जिसको हम बाहर से देखते हैं, जिसको हम ऑब्जर्व करते हैं, ऑब्जर्वेशनल रिसर्च, तो वो जो है हमारी, जिसमें हमने कुछ करना नहीं, खाली ऑब्जर्व करके अपना जो है डाटा निकालना, ऑब्जर्व करके हमने फीडबैक देना है, ऑब्जर्व करके हमने फैक्ट निकालने हैं। तो उसमें जो है हमारी आ जाती है आपकी एक्सप्लोरेटरी रिसर्च, डिस्क्रिप्टिव रिसर्च, कोरिलेशन रिसर्च, एक्सप्लेनेटरी रिसर्च। और जहां पे हमने कोई काम करना है, रियल में किसी को दवाई दी और दवाई करके वो दवाई काम करती है नहीं करती है। कोरोना में हमने जो है अपना सुदर्शन चूर्ण दिया और वो काम करता है नहीं करता है, तो एक ये एक्सपेरिमेंट वर्णन करके, किसी को जो है रिलेशन बैठा के, किसी को एक्सप्लेन करके, आपने जो है नई नई चीजों को सामने लाके जो रिसर्च करते हैं, वो ऑब्जर्वेशन में आती है, जो एक्सपेरिमेंट में। इसी तरह रिसर्च दो तरह की होती है, प्राइमरी रिसर्च, सेकेंडरी रिसर्च। प्राइमरी रिसर्च माने जो वास्तव में मूल रूप से किसी व्यक्ति या किसी रिसर्चर के द्वारा की गई हो, वो प्राइमरी रिसर्च कहलाती है। और सेकेंडरी रिसर्च, जो रिसर्च किसी प्राइमरी वर्कर ने करी और उस रिसर्च को या उस जैसे 10 रिसर्चर के डाटा को रिव्यू करके फैक्ट को, कुछ सिमिलरिटी को, कुछ विशेषता को, सामान्यम चा विशेषम चा द्रव्य गुण कर्माण चा, उसको सर्वदा सर्वभावानाम सामान्य वृद्धि कारणम, इस भावना से उसको इवेल्युएट करना, विश्लेषण करना, ये जो है रिसर्च का काम है इसका। अब रिसर्च की अगर हम बात करें तो रिसर्च से पहले जो है मैं और शेयर करना चाहता हूँ कि रिसर्च की कुछ बेसिक बातें हैं। अगर रिसर्च की डेफिनेशन आप पढ़ेंगे, रिसर्च जो है शब्दों से बना, पहले आर का मतलब होता है रैशनल वे ऑफ थिंकिंग, आपका तार्किक चिंतन अगर आप करते हो तो रिसर्च आप करते हो। आप ये नोट कर लीजिए सारे लोग। ई फॉर एक्सपर्ट एंड एग्जॉस्टिव ट्रीटमेंट, जब आप जो है एक्सपर्ट, विशेषज्ञ लोग अगर कुछ काम करें, मेहनत के साथ काम करें, तो डेट इज ई। सेकेंड आता है आर ई एस, एस फॉर हमेशा समाधान के लिए खोज होती है, समाधान के लिए मेहनत करी जाती है, तो सर्चिंग फॉर सॉल्यूशन। लास्ट में जो है एस के बाद फिर ई आता है, इसका मतलब होता है शुद्धता, एग्जैक्टनेस, क्लियरिटी, एक्यूरेसी। जब भी अगर हम रिसर्च में अगर ये नहीं होगा जो मैं करेक्टरिस्टिक बता रहा हूं, तो आप मान लीजिए कहीं ना कहीं गलतियां हो रही। इसी तरह फिर आता है हमारा ए फॉर एनालिटिकल व्यू एंड एनालिटिकल डाटा, जहां पे एनालिसिस हो, जहां पे विश्लेषण हो, जहां पे डाटा का विश्लेषण हो, वो सारे के सारे आपके एनालिसिस में आते हैं। इसी तरह फिर नेक्स्ट एस ई ए आर, आर के बाद आता है
Kindly unmute yourself.
जिसमें तथ्यों के साथ आपका जो है सामंजस्य हो, तथ्यों को सामने रिलेशन के साथ रखा गया हो, तो ये आर का मतलब होता है। नेक्स्ट होता है सी, जिसमें कि कंस्ट्रक्टिव अप्रोच हो, कंस्ट्रक्टिव एटीट्यूड हो, कोई शोध, कोई चीज निकालनी हो, कोई चीज जो है कलेक्ट करनी हो। इसके अलावा एच फॉर हॉनेस्टी एंड हार्ड वर्किंग। तो दीज आर द बेसिक कैरेक्टरिस्टिक ऑफ रिसर्च। और रिसर्च की एक अपनी प्रोसेस होती है। ये आयुर्वेदिक पद्धति है। मॉडर्न पद्धति क्या है, पहले मॉडर्न पद्धति बताता हूँ, फिर आयुर्वेदिक पद्धति बताता हूँ। आयुर्वेदिक पद्धति क्या है कि हम 5 प्रकार के अवयव, 5 प्रकार की बातों से हम चीजों का पता करते हैं, स्थापना करते हैं। इसके लिए सबसे पहले हम प्रतिज्ञा करते हैं। सबसे पहले हमें कोई परेशानी होती है, या हमें तो या तो ज्ञान होना होता है, या हमें कोई संकट का समाधान करना। उसके लिए हम रफ्ट अप, एक जिसका कोई आधार नहीं, जिसका पर हम एक इमेजिनेशन के द्वारा एक सॉल्यूशन खोजते हैं, प्रीलिमिनरी सॉल्यूशन, जिसको कि अभी टेस्ट करना बाकी है, वो प्रतिज्ञा कहलाती है। प्रतिज्ञा को या तो स्थापना या तो उसको स्थापित करते हैं बाय द एक्सेप्टिंग द अल्टरनेटिव हाइपोथेसिस और रिजेक्टिंग द नल हाइपोथेसिस, और प्रतिस्थापना। तो इसमें क्या होता है कि या तो प्रतिस्थापना में एक्सेप्टिंग दी नल हाइपोथेसिस और रिजेक्टिंग दी अल्टरनेटिव हाइपोथेसिस। सेकंड क्या रहता है इसमें, हेतु। हेतु हेतु इज द ओल्ड रिसर्च टूल। आजकल जैसे जितनी भी रिसर्च माइथोलॉजी है, हमारे ट्रेडिशनल टाइम में रिसर्च टूल हुआ करते थे। प्रत्यक्ष प्रमाण, अनुमान प्रमाण कहते हैं। प्रत्यक्ष किम प्रमाणम, जो सामने दिख रहा है अगर वो है तो फिर हमें प्रमाण की जरूरत नहीं। इसी को एविडेंस बेस्ड मेडिसिन के रूप में आज सारी दुनिया स्वीकार कर रही है। और आयुर्वेद को आज के समय की आवश्यकता है कि प्रत्यक्ष के पैरामीटर पर खरा उतार के ही हम अपनी सारे लोगों में अपने को पा सकते हैं, पुनः अपने गौरव को स्थापित कर सकते हैं। इसी तरह जो है हम ऐतीय प्रमाण, अपने उपमान प्रमाण, सिमिलरिटी के द्वारा हम जो है हेतुओं को सिद्ध करते हैं। हमारे यहाँ तीसरा टूल था दृष्टांत, सपोर्टिव थेरेपी, एग्जाम्पल्स। एग्जाम्पल्स के द्वारा पुराने समय में जो हमारा रिसर्च या समझाने का तरीका था वो दार्शनिक था। हमारे यहाँ कोई एक्सपेरिमेंटल ट्रायल, क्लिनिकल ट्रायल बहुत सारे इतने मास में नहीं हुआ करते थे। हमारे यहाँ एक अलग टेक्नोलॉजी थी, लेकिन फिर भी ट्रायल होते थे। पुराने जमाने में दे दिया तो केमिकल एनालिसिस कर लिया, फ्लेम टेस्ट कर लिया, उसकी विष परीक्षा कर लेते थे। फिर उसके बाद उसको हम जो है जानवरों को खिलाने का विधान था, एनिमल ट्रायल कहीं ना कहीं थे। फिर ह्यूमन ट्रायल, मानव में भी प्रयोग करते थे, लेकिन वो एक दृष्टांत के द्वारा दार्शनिक तरीके से ज्यादा उसको प्रमोट करते थे, बताते थे। क्योंकि लोगों की एक सोच या जो है प्रमाण वाली नहीं थी, प्रत्यक्ष वाली नहीं थी। उनको एक कंसेप्चुअल चीजों को अगर बता दिया जाता था तो लोगों को समझ में आता था। तो इसी तरह उपनयन, अगर जब हम जो है किसी जो फैक्ट्स हैं उनको, हाइपोथेसिस है उनको हेतुओं के द्वारा, दृष्टांत के द्वारा जब बताते हैं, तो ये उपनयन के द्वारा हम निगमन करते हैं, कंक्लूजन करते हैं, एस्टेब्लिशमेंट करते हैं। और लगभग यही सारी क्रिया हमारे एलोपैथिक सिस्टम में भी या मॉडर्न सिस्टम में भी होती है। एलोपैथिक के माने, ये रिसर्च बहुत छोटा टर्म हो जाएगा, ये माने पूरे के पूरे कोई भी विज्ञान है उसमें जो है यही तरीका होता है। क्या तरीका होता है? देयर आर द 5 स्टेप। पहला स्टेप होता है परसीव द क्वेश्चन। क्वेश्चन चाहे ज्ञान की पिपासा हो, चाहे संकट का समाधान हो, उन संकट के समाधान या ज्ञान की पिपासा के लिए परसीव द क्वेश्चन। क्वेश्चन करने के और बनाने के भी अपने तरीके हैं, बहुत बड़ी माइथोलॉजी के लेक्चर हैं उसके लिए। फिर क्या है? फॉर्मुलेशन दी हाइपोथेसिस। उसके समाधान के लिए आपने एक हाइपोथेसिस का एक निर्माण करना, एक प्रीलिमिनरी, एक आपने आइडिया, एक सॉल्यूशन खोजना, ये हाइपोथेसिस। तो टेस्ट दी सॉल्यूशन, सॉल्यूशन को जो है किसी भी हाइपोथेसिस के द्वारा, नल हाइपोथेसिस, अल्टरनेट हाइपोथेसिस के द्वारा, वेरियस एक्सपेरिमेंटल टूल के द्वारा उसको परखना, जांचना और देखना, और उसके द्वारा जो निष्कर्ष निकलते हैं उनके रिजल्ट को देखना, ऑब्जर्व करना, एनालिसिस करना, टू ड्रा द कलेक्शन, कंक्लूजन। तो इसको हम कहते हैं परिकल्पना का निर्माण, समस्या का निर्धारण, आपका परीक्षण, परिकल्पना का परीक्षण, और निर्णय, निष्कर्ष, और रिपोर्ट राइटिंग। जो भी निष्कर्ष निकला उसको, ये सारे के सारे चीजें हैं। और हमारे यहाँ रिसर्च के लिए कहा गया कि रिसर्च जो है
Hello.
तो कहने का मतलब जो है इथिकल कोरिलेशन भी है, इथिकल रिलीवेंस भी होनी चाहिए, एक्सेप्टेबिलिटी भी होनी चाहिए कि हम कोई नशा वाली चीज ना बनाए, हम कोई जो है समाज को बिगाड़ने वाली चीज ना बनाए। तो इथिकल एक्सेप्टेबल हो और रिलीवेंस हो और उसकी समय के अनुकूल मांग हो। पता लगा जो है हम 20 साल पुराने 50 साल पुराने विषय पे जिस पे बहुत काम हो चुका और उस पे काम करेंगे तो वो गलत होगा ना। इसी तरह जो है फिर आता है रिसर्च टॉपिक कैसा हो, ऑब्जेक्टिव क्या हो। रिसर्च का जो है ऑब्जेक्टिव मूल तो मैंने बता दिया कैसे होते हैं कि या तो ज्ञान की खोज, ज्ञान की पिपासा को शांत करना या संकट को दूर करना, प्रॉब्लम को दूर करना। फिर इसके लिए कहा गया कि रिसर्च के ऑब्जेक्टिव जो है माइथोलॉजी में कहा गया कि स्मार्ट होने चाहिए। स्मार्ट क्यों होने चाहिए? क्योंकि स्मार्ट कोई नहीं होगा तो टिकेगा ही नहीं, वो जो है खरा नहीं उतरेगा, उसके रिजल्ट नहीं आएंगे। तो क्या होना चाहिए ऑब्जेक्टिव? स्पेसिफिक होने चाहिए, पर्टिकुलर, कि हमने जो है कोरोना में ये देखना है कि इसका सीआरपी कम होता है कि नहीं होता है, इसका जो है डी डाइमर का क्या रिजल्ट है, इसमें जो है ब्लड के जो है जनरल ब्लड पिक्चर का क्या रोल है। मतलब पर्टिकुलर जो है अगर हम स्पेसिफिक ऑब्जेक्ट के साथ अगर काम करेंगे तो समाज में स्वीकारता रहेगी। इसी तरह मीजरेबल, हमने जो है जो भी काम करें उसको नापा जा सकता हो, उसको मीजर करा जा सकता हो, उसका मात्रात्मक रूप से विश्लेषण किया जा सकता हो। ऐसे अचीवेबल, ऐसे ऑब्जेक्ट लेने हैं जिसको प्राप्त कर सकते हो। जैसे अगर मैं जो है ऑब्जेक्टिव ले लूं कि सिटी वैल्यू जो है 25 से ज्यादा है और उन पे सब पे मैं जो है काम करूंगा, तो आपने धरातल पे रह के अपने ऑब्जेक्टिव को डिफाइन करिए। सर अभी टाइम है हमारे पास, सॉरी। सर अभी हमारे पास कितना समय होगा सर? इसी तरह जो है हमारे ऑब्जेक्टिव अचीवेबल होने चाहिए, टाइम बाउंड होने चाहिए, कि जिसको एक समय पे पूरा कर सकें। ऐसा नहीं कि आपने रिसर्च ले ली 20 साल तक आपकी रिसर्च चली रही, आपका जो है एक टाइम बाउंड मैनर में कोई भी काम होना चाहिए। तो इसी तरह जो हाइपोथेसिस के बारे में बताया गया, हाइपोथेसिस सिंपल होनी चाहिए, स्पेसिफिक होनी चाहिए, कैपेबल फॉर बीइंग टेस्टिंग, जिसको कि टेस्ट कर सकते हो, स्टेटेड इन एडवांस, पहले से हमने लिखा होना चाहिए, बता देना चाहिए, उसको जो है प्रेजेंट टेंस में, प्रेजेंट उस काल में करते हैं। और ये दो तरह से होती है हाइपोथेसिस, नल दो तरह से होती है, वन साइडेड हाइपोथेसिस, डायरेक्शनल हाइपोथेसिस, अनडायरेक्शनल और टू साइडेड हाइपोथेसिस। अब फिर बात आती है कि आरएनडी की जरूरत हमें क्यों है। आरएनडी में क्या क्या इंफ्रास्ट्रक्चर लगते हैं एक अच्छा सेटअप में, तो उसके लिए जो है 5 प्रकार के एम होते हैं, 5 एम। मैन के रूप में साइंटिस्ट, रिसर्च एसोसिएट, प्रोफेसर, साइंटिस्ट, एफ, बड़े बड़े पूरा स्टाफ चाहिए जिसमें एनालिसिस से लेकर लैब, केमिस्ट और पूरा एक इंफ्रास्ट्रक्चर ऑफिस। इसी तरह मटेरियल में, रिसर्च इनपुट्स में सारे हमें जितने भी जो है मटेरियल्स, जितने भी रिएजेंट हैं, जितने भी जो है टेस्टिंग के लिए जितने भी रिक्वायर्ड इनपुट्स हैं वो सारे के सारे हमें चाहिए। इसी तरह मशीन्स में आरएनडी की सारी मशीन्स, इक्विपमेंट्स, मेथड में स्टडी डिजाइन, एसओपीज, वर्क इंस्ट्रक्शन, एसटीपीज, क्रिटिकल कंट्रोल पॉइंट आरएनडी के लिए चाहिए। और मनी और रिकरिंग कैपिटल में वर्किंग कैपिटल मशीनों को खरीदने के लिए। और तो आरएनडी का क्यों हमें आवश्यकता है आज के परिप्रेक्ष्य में? तो आरएनडी की आवश्यकता हमें इसलिए है क्योंकि हम इससे नए दवाइयों को खोज सकते हैं। नई नई विधाओं को खोज सकते हैं, नई नई समस्याओं के सॉल्यूशन को हम ला सकते हैं दुनिया के सामने। इसी तरह जो है, अगर हमने नई दवाइयों के रास्ते खोजने हो, कैसे दवाई बॉडी के अंदर प्रवेश करती है, टू इन्वेंट दी न्यू ड्रग डिलीवरी सिस्टम। ब्लड ब्रेन बैरियर से दवाइयां पार नहीं होती है, मोस्टली दवाइयां पार्टिकल को बनाने की कलाएं हैं, तो उनके द्वारा हम नए नए सिस्टम को खोज सकते हैं। टू हाईएस्ट एफिकेसी, ऐसी दवाइयों का निर्माण करना जिनकी एफिकेसी हो, दवाई बन गई है लेकिन दवाइयों को और एफिकेशियस बनाना। दवाई है लेकिन उसकी सेफ्टी को बढ़ाना, दवाई जो है प्रूव करना, अप्लाइड रिसर्च करना, ये हमारे जो है प्रोडक्ट के जो एग्जिस्टिंग एट्रीब्यूट हैं, जो अभी हमारे डॉक्टर मांझी ने बताया कि कैसे कैसे पहले मोबाइल क्या था, आज की डेट में क्या आ गया। तो प्रोडक्ट के जो भी एट्रीब्यूट है, चाहे उसकी अपीयरेंस हो, चाहे उसके टेस्ट हो, शेप हो, उसकी स्मेल हो, उसकी प्राइस हो, सब एट्रीब्यूट को हम ज्यादा से ज्यादा अच्छा बनाना। तो प्रोडक्ट के एट्रीब्यूट में आते हैं प्लांट सोर्स है, जियोग्राफिकल एनवायरनमेंट है, प्लांटिंग टेक्नोलॉजी है, प्लांट हार्वेस्ट टाइम है, मेडिसिनल साइट है, मार्केटिंग, रॉ मटेरियल्स, प्रोसेसिंग टेक्नोलॉजी, मैन्युफैक्चरिंग प्रोसेसेस, प्रिवेंशन, प्रिजर्वेटिव जो प्रिजर्व करने के लिए जो सारी बातें हैं, ड्रग रेगुलेशन एप्लीकेशन। तो इतनी सारी बातें हैं जिस पे हमें रिसर्च के लिए कुछ ना कुछ जरूरत पड़ती है। लेकिन अगर ओवरऑल हम अगर रिव्यू करें तो रिसर्च जो है दो तरह से होती है, प्राइमरी रिसर्च, सेकेंडरी रिसर्च। प्राइमरी रिसर्च जो कि हमने डायरेक्ट खुद करी हो, सेकेंडरी रिसर्च जो हमने किया है उसको रिव्यू एनालिसिस किया हो। तो प्राइमरी रिसर्च को बेसिक रिसर्च, जिसमें कि हम जो है फंडामेंटल रिसर्च के द्वारा दो तरह से करते हैं, थियोरेटिकल रिसर्च या अप्लाइड रिसर्च। और क्लिनिकल रिसर्च को हम फिर दो तरह से करते हैं, एक्सपेरिमेंटल रिसर्च और क्लिनिकल रिसर्च। इसमें एक एक्सपेरिमेंटल आ जाता है और एक ऑब्जर्वेशनल, कि किसी ने कुछ किया और उसके आधार पे हमने अपने आप को रिव्यू करके उसको नोट कर लिया। इसी तरह एपिडेमियोलॉजिकल, जिसमें कि हम जो है जितने भी वातावरण के जितने भी रोग हैं, कम्युनिकेबल डिजीज हैं, इनको एक्सपेरिमेंट या ऑब्जर्वेशन के द्वारा इनको रिव्यू करना या इनको आइडेंटिफाई करना, इनके रिजल्ट को। इसी तरह सेकेंडरी रिसर्च में मेटा एनालिसिस और जो है रिव्यू, और इनमें सिस्टमेटिक और सिंपल रिव्यू के द्वारा हम चीजों को करते हैं। एक हमारे यहाँ रिसर्च की और एक धारा है लिटरेचर रिसर्च, जिसमें कि हम वांग्मय को खोजते हैं। लिटरेचर रिसर्च दो तरह से होती है, आयुर्वेदिक रिसर्च के लिए, नॉन आयुर्वेदिक रिसर्च के लिए। इसमें दो कैटेगरी रहती है, पब्लिश, अनपब्लिश। और फिर इसमें भी और कैटेगरी रहती है कि देश में पब्लिश, विदेश में पब्लिश। तो इनमें जो है हम पांडुलिपियों, रिसर्च जर्नल, विभिन्न प्रकार के जो है रिसर्च के आर्टिकल हैं, बुक्स हैं, इन सब के द्वारा जो है थीसिस है, डिसर्टेशन है, इनके द्वारा हम खोज करते हैं। ड्रग रिसर्च, ड्रग रिसर्च आज की डेट में सबसे महत्वपूर्ण पहलू है, क्योंकि दवाइयों का कहते हैं विषक द्रव्याणु उपस्थाता रोगी पाद चतुष्टयम, ये चार पाद हैं जिसमें कि वैद्य यद्यपि प्रधान है, लेकिन वैद्य भी किसके अधीन है, वो दवाइयों के अधीन है। दवाई के लिए बताया गया संपन्नम योग्य औषधम, अगर दवाई संपन्न होगी, योग्य होगी और उसमें सारे गुण होंगे, उसकी जो है चॉइस होगी आपके पास, तो दवाई आपके लिए, लोगों के जन कल्याण के लिए वो लाभ करेगी। तो दवाई के लिए एक सिंपल ओवरव्यू आप समझ सकते हैं कि सबसे पहले कोई भी रिसर्च होती है तो कहाँ से शुरू होती है, सबसे पहले खोज से शुरू होती है। उसके लिए मार्केट क्रिएट होना चाहिए। मार्केट माने बेचना नहीं होता, मार्केट माने होता है कि लोगों की नीड को करना, सेल माने होता है बेचना। मार्केट माने क्या होता है कि लोगों को किसी के लिए जरूरत के लिए तैयार करना, मेक पीपल एक्सेप्टेबल फॉर द प्रोडक्ट, दैट इज कॉल्ड मार्केटिंग। और सेल माने क्या होता है, बेचना। तो इनमें क्या करते हैं कि हम समय के अनुकूल प्रॉब्लम के आधार पर पहले एक खोज करते हैं कि हमें किस चीज पे काम करना है, और उस काम के लिए हम उसकी लिटरेचर रिव्यू करते हैं, उसका एग्जिस्टिंग जो है प्राइमरी सेकेंडरी डाटा जो भी अवेलेबल है सारी दुनिया में, उसको देखने के बाद हम स्टेटस देखते हैं आज तक इस पे क्या काम हुआ है, और उस काम के बाद फिर हम जो है उसके लिए हम एक हाइपोथेसिस बनाते हैं कि मान के चलिए कोरोना है, कोरोना में इससे पहले चरक ने तो हालांकि लिखा है कि जो इस चरक में है वो सब जगह है और जो चरक में नहीं वो कहीं भी नहीं। और चरक ने ये भी लिखा कि जो है जहां भी आपको वक्त के साथ साथ व्याधियां, इनके स्वरूप, इनके नाम बदल जाएंगे, लेकिन बात पित्त कफ वही रहने वाले हैं, और इन्हीं सिद्धांतों से आप इनकी आगे आप अपना काम चलाने वाले हैं। तो अब जो है जब अगर कोई व्याधि आई है सामने, तो उसके लिए हमने अपने आप को रिव्यू करा, माइंड सेट करा कि भाई इसमें जो है कोरोना में ये ज्वर होता है, तो इसमें पित्त का प्रकोप है, इसमें जो है ये खांसी के लक्षण आ रहे हैं, कफ के लक्षण आ रहे हैं, तो बात कफत जो है व्याधि है। तो बात कफत के लिए कौन कौन से हैं? त्रिवन कीर्ति रस हो सकता है, हमारा जो है जो भी हमारी औषधियों के जो रेजिम बताए हैं, जो हमारे जो है चाहे महासुदर्शन चूर्ण हो, चाहे जो है संशमनी बटी हो, या किसी भी तरह की जो औषधियां बताई गई हैं, तो एक प्रीलिमिनरी हमने एक माइंड सेट किया, और माइंड सेट करने के बाद हमने एक कुछ एग्जिस्टिंग दवाइयों में से कुछ जो है रॉ मटेरियल को हमने सिलेक्ट करके एक जो है रेशनल तैयार करा। जो मैंने रिसर्च की परिभाषा बताई, क्या बताई है? रेशनल वे ऑफ थिंकिंग। तो एक रेशनल के द्वारा हमने एक फॉर्मुलेशन को डिजाइन करा, और डिजाइन करके उसके द्वारा एक प्रीलिमिनरी हमने औषधि का निर्माण किया। औषधि को निर्माण करके उसको हमने एनालिसिस करा, उसको स्टैंडर्डाइज करा, उसको फॉर्मुलेशन में एफएनडी में उसको जो है पूरा स्टैंडर्डाइज करने के बाद एनालिसिस केमिस्ट्री में डाला। उसकी जो है अभी फर्दर मैं आपको शेयर करता हूँ। हमने क्या किया? ऑर्गेनोलेप्टिक टेस्टिंग के द्वारा उसको जो है पंचेंद्रिय के द्वारा उसको परीक्षित किया कि उसका रंग कैसा है, उसका टेस्ट कैसा है, उसकी गंध कैसी है। उसको बॉटनिकल कैरेक्टरिस्टिक के द्वारा, मैक्रोस्कोपिक और माइक्रोस्कोपिक कैरेक्टरिस्टिक के द्वारा, फिर फिजिको केमिकल जो कैरेक्टरिस्टिक होते हैं, चाहे आपका जो है मॉइस्चर कंटेंट हो, चाहे एक्सट्रैक्टिव वैल्यू हो, चाहे वाटर सॉल्युबल एक्सट्रैक्टिव वैल्यू हो, एस वैल्यू है, दुनिया भर के एसेज हैं। इसी तरह उनके सेफ्टी प्रोफाइल के लिए माइक्रोबियल कंटामिनेशन को देखा हमने, बैक्टीरियल लोड को देखा, फंगल लोड को देखा, 4 तरह के पैथोजन को देखा। इसी तरह केमिकल एनालिसिस के लिए विभिन्न प्रकार के ऐसे क्वालिटेटिव क्वांटिटेटिव फिंगरप्रिंटिंग, एचपीटीएलसी फिंगरप्रिंटिंग, और हैवी मेटल पेस्टिसाइड रेसिड्यू और एफ्लाटॉक्सिन, तो इनके आधार पर हम सेफ्टी की सारी परीक्षा करते हैं। और इसमें हम एक लमसम एक प्रोफाइल बना लेते हैं, जिसके आधार पर हम जो है एक सेफ्टी को जनरेट करते हैं। सेफ्टी स्टडी के बाद, जो कि हम लॉन्ग टर्म शॉर्ट टर्म सेफ्टी के बाद हम ये इंश्योर हो जाते हैं कि इससे किसी को नुकसान नहीं होने वाला। इसकी आयुर्वेद की या किसी भी साइंस की ब्यूटी ये है कि बहुत ज्यादा इथिकल कंसंट्रेशन है। जो मैंने बताया ना कि इथिकल कंसिडरेशन रहना चाहिए हमारी रिसर्च में, तो उसके द्वारा हम जो है प्री लॉन्ग टर्म और शॉर्ट टर्म स्टेबिलिटी के द्वारा जब हम संतुष्ट हो जाते हैं, तो हम प्री क्लिनिकल स्टेज में जाते हैं। प्री क्लिनिकल में हम दवाइयों का एक जो है किसी भी मॉडल के आधार पे, जैसे अगर हमने च्यवनप्राश का रोल देखना है, तो फ्री रेडिकल स्कैवेंजिंग एक्टिविटीज के द्वारा हमने देखा कि उसमें कैसे आंवला काम कर रहा है, कैसे गिलोय काम कर रहा है, कैसे उसमें MLN2 एंटीऑक्सीडेंट का काम कर रहा है। तो इनके जब प्री क्लिनिकल से स्क्रीनिंग से हमें कुछ रिजल्ट मिल जाते हैं, फिर हम इस पे आते हैं कि ये कैसे काम कर रहा है ये। तो उसके लिए एक हाइपोथेसिस बनाते हैं। उसके लिए हम या तो बातचीत कप के आधार पर सोचते हैं, या इसके मेडिकल मार्कर के आधार पर हम एक निर्धारण करते हैं, और फिर जो है हम क्लिनिकल ट्रायल में जाते हैं। और इसके लिए जो एक सारा का सारा जो स्टैंडर्डाइजेशन का प्रोसेस है, उसको हम 4 बिंदुओं में बांट सकते हैं। इनपुट्स, जिसमें कि वैलिडेटेड प्रोसीजर, चाहे वो AFI से आए चाहे API से आए, जिसमें विभिन्न प्रकार के SOPs होते हैं, फॉर्मुलरीज होती हैं। वैलिडेटेड इक्विपमेंट, जिसमें कि PQ, IQ, DQ और OQ को हम वैलिडेट करते हैं। ट्रेन टेक्निकल पर्सनल, जिसको कि GMP और OSD और GCP की हम ट्रेनिंग दिलाते हैं। और वेरियस प्रकार के रेफरेंस स्टैंडर्ड, जिनको CRM बोलते हैं, जो NABL टेस्टिंग के लिए आवश्यक होते हैं। और स्पेसिफिकेशन, तो इनके द्वारा जो है हम प्रोसीजर को इकट्ठा करते हैं, लेबोरेटरी मैनुअल बनाते हैं, टेस्टिंग मैनुअल बनाते हैं, PMR क्रिटिकल कंट्रोल पॉइंट डिसाइड करते हैं, STP बनाते हैं, स्टैंडर्ड टेस्टिंग प्रोसीजर, वर्किंग SOPs बनाते हैं, वर्क इंस्ट्रक्शन बनाते हैं, और इनको हम मेंटेन करते हैं कैसे? स्टैंडर्डाइजेशन के द्वारा। और इसको हम पर्टिकुलर कैसे मेंटेन करते हैं? कैलिब्रेशन के द्वारा, प्रोफिशिएंसी टेस्टिंग के द्वारा। और फिर जो है इससे हमें आउटपुट क्या मिलता है? टेस्ट रिजल्ट मिलता है, वैलिडेटेड डॉक्यूमेंट मिलता है, जिसके आधार पर ड्रग जब स्टैंडर्डाइज होता है, तो फिर हम इसको क्लिनिकल ट्रायल में डालते हैं। तो इसमें हम स्टेबिलिटी स्टडी भी करते हैं। तो आयुर्वेद में जो स्टेबिलिटी स्टडी जो चलती है, वो रियल टाइम स्टेबिलिटी स्टडी चलती है, और ये सारी हमारी एक्सेप्टेबल रेंज होती है प्रोडक्ट की। अब नाउ कम ऑन दी जनरल डिफिकल्टीज ऑफ दी R&D एक्टिविटीज। आप सभी जानते हैं कि R&D के बारे में हमारे सारे वरिष्ठ जनों ने लगभग सारे एस्पेक्ट पे जो है बात कर ली है। तो लेकिन R&D में बहुत कुछ होने के बावजूद, वैसे मेरा तो ये मानना है कि रिसर्च का मतलब क्या है, दो तरह के लोग होते हैं, ऋषि और मुनि। जो रिसर्च कर रहे हैं वो ऋषि, और जो मनन कर रहे हैं, अध्ययन कर रहे हैं, वो मुनि। तो आज की डेट में ऋषियों की जरूरत है, मुनि तो बहुत हैं, ऋषियों की जरूरत है जो रिसर्च कर रहे हैं। तो रिसर्च का मेरे शब्दों में मतलब ये है, री एस्टेब्लिशमेंट ऑफ ओल्ड फैक्ट थ्रू मॉडर्न माइथोलॉजी। आज की ये समय की मांग है कि टाइम टेस्ट से हमारे पास, चाहे ट्रांसप्लांट से लेके सारी चीजें हमारे पास चिकित्सा विधाओं में भरी पड़ी हैं, दसियों हजार फॉर्मुलेशन भरे पड़े हैं, लेकिन आज को जो है साइंटिफिक लैंग्वेज में लोगों को साइंटिफिक तरीके से वैलिडेट करते हुए समझाने के लिए हमारे पास डॉक्यूमेंट चाहिए, एविडेंस चाहिए, रिसर्च पेपर्स चाहिए, उनकी जो है कहानी चाहिए। तो उसके लिए हमारे पास लैक ऑफ स्किल्ड पर्सन नहीं है। ऐसे लोग तो हैं जिन्हें आयुर्वेद आती है, लेकिन ऐसे लोग नहीं है जिन्हें साइंस आती है। साइंस और आयुर्वेद में थोड़ा अंतर है। तो आयुर्वेद को हमें साइंटिफिक वैद्य बनाने हैं आज की डेट में, जिनका जो है ट्रेडिशन विद टेक्नोलॉजी, जिनको खबर हो। इसी तरह इंस्ट्रूमेंट जो विशेष है, सेटअप है R&D का, बहुत कॉस्टली है। आज की डेट में जो है हम सिंपल टेस्टिंग भी उस लेवल पे नहीं कर पाते हैं, लेकिन जो है अगर मॉडर्न के ड्रग देखें तो अरबों खरबों डॉलर जो है खर्च हो जाते हैं। तो इसने हमारे पास ना तो प्रमोटर होते हैं ना स्पॉन्सर होते हैं। डेट इज द बिग प्रॉब्लम फॉर आयुर्वेदिक पर्सन। आप अपने इंडिया में देखें 10,000 आयुर्वेदिक फार्मेसी हैं लगभग, और एलोपैथी में भी लगभग इतनी है, 10,000 के लगभग। तो उसमें से सिर्फ जो है 5, 7, 10 लोगों के पास एक अच्छी R&D होगी, वरना मुझे लगता है खाली दिखाने के लिए R&D होगी, लेकिन रियल में R&D क्या होती है ये मतलब बहुत बड़ा चिंता का विषय है। लैक ऑफ रिटेन डॉक्यूमेंट्री एविडेंस ऑन द क्लिनिकल एफिकेसी। आज की डेट में बहुत ज्यादा हमारे पास रिटेन एविडेंस नहीं है, तो हमें जो है सारा का सारा फिर से एक नए सिरे से काम करना पड़ेगा। हमारे पास SOPs नहीं है, विभिन्न प्रकार की डाइवर्सिटी, जैसे अश्वगंधा है, अगर एक आपकी नागौर की काम करती है, पहाड़ की अश्वगंधा उतनी है काम नहीं करती। इसी तरह आंवला जो है प्रतापगढ़ का अच्छा होता है, तो जो बसनाव है वो पहाड़ पे ऊपर जितनी हाइट पे होगा उतना अच्छा होता है। तो इसी तरह जो है अगर बादशाह है तो अगर बसंत ऋतु में लेंगे तो उसका रोल अच्छा है। तो किसी का समय वय काल के हिसाब से रिजल्ट अलग अलग हो जाते हैं, और इन सब के लिए हमारे पास बहुत ज्यादा इंफॉर्मेशन नहीं है, और है तो वो रिटन फॉर्म में साइंटिफिक तरीके से नहीं है, तो उसको प्रस्तुत करने की जरूरत है। इसी तरह वैलिडेटेड डॉक्यूमेंट, और ये आपको शॉर्ट में बता देता हूँ, टाइम बहुत ज्यादा हो गया है ऑलरेडी। तो ये सब जितने भी पासपोर्ट डाटा है, हमारे टॉक्सिसिटी प्रोफाइल है, इन सबको बहुत ज्यादा हमें कलेक्ट करने की जरूरत है। अब इसको मैं समराइज करना चाहूँ तो यही कहूंगा कि जो है आज की डेट में क्योंकि आयुर्वेद में स्कोप की कमी नहीं, सारी दुनिया भाग रही है, और कोरोना के बाद तो मुझे लगता है कि सारे रास्ते दरवाजे जो बंद थे वो मैं कहना चाहता हूँ आप सभी को खुल गए। हम लोगों के लिए जैसे बाढ़ आती है तो डैम के रास्ते जो बंद होते हैं खुल जाते हैं, तो अब जो है फोर्सफुली हम अपने आप को एक्सपेंड कर सकते हैं, फैला सकते हैं। बस एक अच्छी साइंटिफिक मेथोलॉजी की जरूरत है। मेथड और मेथोलॉजी में यही अंतर है कि जो मेथड होता है जिसमें ये पर्टिकुलर विधि की बात होती है, लेकिन जहाँ पे GLOGY लग जाता है तो वहां पे विज्ञान बन जाता है। चाहे टर्मिनोलॉजी हो, चाहे जूलॉजी हो, चाहे आपका जो है कोई भी आपका लॉजी लगने के बाद वो साइंस बन जाता है। तो मेथोलॉजी के माने ये कि ये विधि नहीं है, ये एक साइंस है, जिसको कि हम कहते हैं शोध प्रविधि, हिंदी में कहेंगे शोध प्रविधि। तो इसको हमें बढ़ाना है, और गवर्नमेंट के लेवल पे कोई ऐसा सेटिस्फैक्ट्री अभी प्रोग्राम मुझे नहीं लगता है, या इंसेंटिव जिसके द्वारा लोग मोटिवेट हों। एक व्यक्ति R&D में पैसे लगा रहा है, इन्वेस्टमेंट कर रहा है, कल को रिजल्ट आएगा नहीं आएगा पता नहीं, लेकिन एक व्यक्ति बेच रहा है उसको तो तुरंत रिजल्ट मिल रहा है, लाभ मिल रहा है। तो इसके लिए इन्वेस्टमेंट, ह्यूज इन्वेस्टमेंट की जरूरत है। तो बहुत सारे, अगर कोई अच्छी R&D करता है तो उसको चाहे टैक्स में बेनिफिट दिलवाए, चाहे जो सब्सिडी दिलवाए, या उसके लिए थोड़ा रिसोर्स अवेलेबल कराए, या गवर्नमेंट के कुछ इंस्टीट्यूट बने जहाँ पे लोग जो है रिसर्च कर पाए, अपने कंसेप्ट को दे पाए, टेक्नोलॉजी ट्रांसफर कर पाए। इस तरह की चीजें डेवलप होनी चाहिए, और जो MOU होने चाहिए यूनिवर्सिटी के साथ, गवर्नमेंट के साथ, इंस्टीट्यूट के साथ, आयुष के वेरियस स्टेक होल्डर्स के साथ, और कोलैबोरेटिव रिसर्च। आयुर्वेद में जो सबसे बड़ी दुर्भाग्य है, मैं अपने लोग बैठे हैं इसलिए शेयर कर देता हूँ कि आयुर्वेद में मतलब छुपाने की बहुत बड़ी एक बीमारी है। मतलब कोई भी अगर हो, अगर कोई नुस्खा अगर मिल गया तो उन्हें ये लगता है कि मेरे साथ ही खत्म हो जाए, मेरे जिंदा रहते रहते रहेगा, लेकिन बाद में नहीं जाएगा किसी को। और शायद इसीलिए आप सारी दुनिया में देखना, कहीं भी जो भी होता है प्रिस्क्रिप्शन में साफ साफ लिखा होता है पैरासिटामोल 500 mg। तो मैं तो अपनी तरफ से कोशिश करता हूँ क्लियर कट जो है प्रिस्क्रिप्शन लिखता हूँ, रेदर देन की दवाई डिस्पेंसिंग का काम जो है पुड़िया बना के कर दिया। लेकिन आयुर्वेद में क्लियरिटी की जरूरत है और कोलैबोरेटिव रिसर्च की, उसमें जो है वेरियस स्ट्रीम के लोगों को, साइंटिस्ट को जोड़ने की जरूरत है। आयुर्वेद में हम लोग क्या समझते हैं, नहीं हम लोग समर्थ हैं, संपूर्ण है, हम तो धन्वंतरी भगवान ने हमें सारी विधाएं दे रखी है, एक हाथ में अमृत कलश दे रखा है, एक हाथ में आयुर्वेद दे रखा है, एक हाथ में जड़ी बूटी दे रखी है। लेकिन आप धन्वंतरी जी को भी देखिए, उन्होंने भगवान होते हुए भी क्या क्या पकड़ रखा है। मेरे सामने ये चेहरा है।
Dr. Rajiv, could you please wind up quickly because
आप
due to our time
कृपा के
running
साथ
late.
खुद भी स्वस्थ है।
Please, sir, please wind up quickly because we are already running behind schedule.
जी जी जी। तो अब मैं
Thank you.
लास्ट बट नॉट लीस्ट आई जस्ट वांट टू शेयर दी। जी 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 जी। तो लास्ट बट नॉट लीस्ट आई वांट टू शेयर सम बेसिक मंत्रा, सक्सेस मंत्रा फॉर गुड R&D सेटअप। अच्छी R&D के लिए क्या चाहिए, एक जो उर्वरा भूमि होती है जिसके लिए ये चीजें हैं जिनके बिना आप R&D नहीं कर पाएंगे और ऐसे उड़ान नहीं भर पाएंगे R&D की या रिसर्च की या खोज की। तो उसके लिए हार्ड वर्क चाहिए, पेशेंस चाहिए। R&D वाले को पेशेंस बहुत ज्यादा चाहिए डॉक्टर साहब, और रिगरस एफर्ट चाहिए, साइंटिफिक टीम वर्क चाहिए विद कोलैबोरेशन ऑफ मल्टी रिसर्च इंस्टीट्यूट अलोंग विद ड्यूटी, 3D और चाहिए, 3D रिसर्च है ये, ड्यूटी, डिवोशन एंड डिसिप्लिन। आप जो है अनुशासन के साथ, पूरी ड्यूटी, पूरी तन्मयता के साथ, पूरे डिवोशन से लगे रहेंगे तो आगे को दे पाएंगे। ऐसा मेरा सभी से जो है विनम्र आग्रह है। थैंक्स फॉर पेशेंस हेयरिंग। बीच में इंटरनेट की डिस्टर्बेंस की वजह से आप सबको 10-15 मिनट बिगड़ रहा, उसके लिए आपसे क्षमा प्रार्थी हूँ। और डॉक्टर पवन शर्मा जी का, डॉक्टर नवीन जोशी जी का और सभी हमारे ऑर्गेनाइजर का मैं हृदय से धन्यवाद देना चाहता हूँ कि मुझ जैसे लर्नर को आपने एक मौका दिया अपनी बात रखने का। थैंक यू सर।
Thank you, Dr. Rajiv, for your nice presentation, and I hope in future we'll get to learn more in details about the research acumen and methodology and the works you are doing in the field of Ayurveda in Uttarakhand University. And we are very thankful to you for giving your time.
Thank you sir. Thank you very much.
Now I would like to request Professor Madan Thangvelu ji from UK to bless us with few words. Dr. Madan Thangvelu ji. Hari Om sir.
Hari Om, Pawan ji, namaskar. Thank you so much for, I have been listening with great interest. Sorry, I couldn't join. Jo shuruaat mein 3-4 jo usme I wasn't able to join, but I could listen very closely to Rajiv ji's presentation, even though there was some problem with the Rajiv ji, aapki wahan Uttarakhand se thoda sa dikkat tha wo connection ke, magar fir bhi humne thoda, I got everything. It's a wonderful presentation. Together with Pawan ji and Joshi ji, what we are, and Vishnu Prasad ji, what we are saying is we need to build a place, build matlab, we must have a base in Uttarakhand. Yahan pe we can establish these things more firmly, you know. I will give you, aapne jo pichli, the last slide jo aapne bataya, R&D wali, you have structured everything well. The issue of costs, let us say, paisa. Ab pura Hindustan ki R&D, pura Hindustan ki health budget, ठीक है ना जी? पूरा देश का health budget, हमारी पूरा देश का health budget is less than the R&D budget, annual research and development budget of the top 3 pharmaceutical companies. ये हाल है हिंदुस्तान, ठीक है ना जी? तो आपने जो बताया चीज, I will send those figures, पवन जी के पास है जो presentation. तो आपने जो बात बताया कि इसमें पैसा की जरूरत है, we need money. What is the formula for this money? So that is one thing, because as soon as we establish a center, Uttarakhand mein, we will establish a center. ठीक है ना जी, ये देश के लिए नहीं, पूरा दुनिया के लिए, ठीक है. So let us think a little more wider. आपने जो बताया, your presentation today has all the details. आपने बहुत बढ़िया, you showed a very nice layout of the research aspects, primary research, secondary research, etc., etc. You know it all. What is the need? What is needed there? What I am suggesting is, let us turn it into action now. Uttarakhand में, आपकी university में, let us present it as a global center for research into Ayurvedic aushadhis, not only aushadhis. Aushadhi to ek hi cheez hai. It is the fourth aspect, and I call the unfortunate aspect of human life. This is the curative aspect. We have three more important aspects where research is needed. And the three aspects are related to prevention of disease. Ayurveda mein, you have the best information about how to prevent disease, in which you have research about Ritucharya, Dinacharya, Swasthurvrit, and the connection with physiology that is sitting there. There is no research going on there, although there is a lot of evidence already. Acha. Second thing is to do with health maintenance. आपके पास इतनी, you have so much information. हमारी संहिताओं में, टीकाओं में इतनी चीज पड़ा, and that is all reinforcing existing understanding of human physiology. अब उसके ऊपर कुछ नहीं हो रहा. We need to consolidate that. The most important one after that is health promotion. कि योग के अंदर, there are so many things that we are meant to do every day. यहाँ पे रिसर्च चल रहा है कि कोविड के ऊपर, कि एडिनबरो में जल नीति से you can control many of the early aspects of COVID. This is jal neti, and that comes from our Yog Darshan. And we are not talking about it. We are just wanting to make कहीं अश्वगंधा ले लिया, कहीं गुरुची ले लिया, उसकी दवाई, उसकी टैबलेट बना के भेजो है. So I don't like that aspect, and we need to open up that agenda. अब हमारी बातचीत चल रहा है एक इसके ऊपर, इरंडे तेल के ऊपर, ठीक है ना जी. इरंडे की that very special Sindhuvaram Iranda. And if I say I am following many cases for intervertebral disc prolapse and backache, the Iranda tail with Sindhuvaram is the best remedy. I'm seeing one case, 7 millimeter stenosis, no din mein. 9th day, the problem is resolved. This is where you can avoid surgery. We need to bring all this together. Ab dekho, I'm asking alag-alag companiyon ke, I'm asking, kiske paas hai ye dawai? Iranda tail, Sindhuvaram Iranda tail. Nobody is making it. Kyun nahi? So let us start putting those examples together and put it in one center, university ke andar, aapke university ke andar, aapke Uttarakhand mein. Let us make this center. Let us give it a name today. This is a global center for, we will establish, we will call it a global center for research. That doesn't cost any money. Just to call yourself a global center, you can, today evening, you can put a name saying this is a global center for research. Uttarakhand Ayurveda University, together with Independent Research Ethics Committee, and just put it on paper. It is sitting in a university, we have students, and this is aapke jo aaj ki presentation wo leke, we just present it. This is our agenda. We want to look at this thing, and we have 20 compounds that we are going to look at. आपने सही से बताया कि प्रतापगढ़ में है आमला की center. This is the world center for amla, but nothing is happening. Amla ki rasayana, standardize the procedure. Kerala mein, there is work going on in amla ki rasayana. Amla ki rasayana for heart disease. It is so important. We are sitting on globally important activities. So my suggestion here, sorry, I'm being a little forceful, because I'm a little emotional because Rajiv ji has presented some very, very valuable things along with the previous speakers. And Pavan ji and Vishnu ji and Joshi ji, all of you have been working together to put this. We are soon approaching the 200th webinar. So I think we must have a very strong, we must have a very strong recommendation for the Ministry of Ayush, for the Ministry of Family Health and Family Welfare in Delhi, very strong recommendations that I'm going to put a very bad word here, but I don't know how to express it better, that you have deceived us till now. This has to stop now. So let us put our minds together. Deception, matlab, we have things here that are working and nobody talks about it. Delhi wale, they don't talk about it. Why is that? So we must be a little aggressive here at this point. So what you have given today, Rajiv ji, is a very, very important lesson for all of us.
Thank you, sir. Thank you. It's my pleasure.
You have put many points together, and let us make this into a set of demands. कि CSIR वालों को पकड़ो, दिल्ली में बैठे डिपार्टमेंट ऑफ बायोटेक्नोलॉजी करके उनको पकड़ो, science and technology wale baithe, I don't know, they don't want to listen. I will send Pavan ji something that I presented at the Center for Soft Power in Delhi. And there are 3 sections there that are very important. I would like all of this to come out with force. Aapne sahi bataya that COVID has opened the dam, opened those barriers now. We must be prepared now to present this. We have concepts, Adhistan of this virus is the colon, is the intestine, and the evidence is there. How can we go into that space? So you have given us a lot of strength today, Rajiv ji, and I want to thank Pavan ji, I want to thank Vishnu Sharma ji, I want to thank everybody there who are holding this platform for all of us to come and express our thoughts, express our wishes, and for people like me to be completely open, apolitical, unpolitical language I'm using here, but there is no other way we can convey our message. We had Director Saab from Shillong who presented the other day. We will bring these people together, and let us make a very substantial recommendation out of the 200 webinars that will come together. We have many things that we can offer India government, and Satyagraha. We will sit there till it happens. I think people in the government in Uttarakhand is very pro Ayurveda. We have many friends there in Uttarakhand who will help us make this happen. And we go looking around the world for research partners. So we won't stop at India. यहाँ जर्मनी में इतनी रिसर्च चल रहा है इरंड तेल की, वो एक फैटी एसिड के ऊपर क्यों काम करती है? What is the magic of इरंड तेल? How does it control अपानवायु? So all of these are things that we should now try to. Vishnu ji gave us an amazing example of how he treats people with complicated spinal inflammation problems. So I feel all of this needs to come out together with Pavan ji, Vishnu ji, and everybody. Let us put our minds together on this, because this is what the world needs today. Poor people of the world are suffering. We need some help from them. So thank you, Pavan ji. Hari Om. Thank you for.
Hari Om. Hari Om. Thank you very much, sir. Thank you very much, sir. Yeah. Now I would like to request Professor Vishnu Prasad Sharma ji to deliver his concluding remarks as blessings.
Okay. Thank you. Namaskar. Respected Professor Madan Thangavalu, Dr. Pavan Kumar Sharma ji, dear distinguished speaker and learned audience. It was a pleasure listening to such insightful talk on Ayurveda Research Methodology GPG series from eminent speaker. Therefore, today's talk on Ayurveda research methodology GP series was significant in several ways. First of all, I offer my sincere thanks to Dr. R.V.B. Prasadji for inaugurating today's webinar. Next, I want to offer my sincere thanks to Professor Suhas Kumar Shetty. He presented newer study design, qualitative and quantitative. He nicely described research study design, type of research, cohort study, case control study, cross-sectional study, animal research very nicely. I offer my sincere thanks to Professor Suhas Kumar Shetty for wonderful talk. Next, I would like to offer my sincere thanks to Dr. A.K. Jain for his nice talk. He nicely described what is research methodology. He nicely described objective of research method and research methodology, various type of research work, experimental research, historical research, research philosophy. And he gives stress on pharmacovigilance and its importance. Nicely described Ayurvedic research, safety and toxicity monitor, method of collection of data, process analysis data, etc. Thank you very much for your wonderful presentation. Next, I would like to give a sincere thanks to Dr. Lieutenant Colonel Dev Kalyan Maji. He nicely described the importance of data collection, data, research methodology, step of research methodology. Thank you, Dr. Lieutenant Colonel Devakalyan Maji, for your wonderful talk. Next, I would like to offer my sincere thanks to Dr. Rajiv Kohli for your excellent presentation. You have nicely described all these things. He has described quality of research, and he nicely described the Ayurvedic procedure. Thank you, Dr. Rajiv Kohli, for your excellent presentation. Namaskar to all.
Thank you.
Thank you.
Thank you, sir, for your kind words. Thank you, sir.
Okay, thank you. Namaskar, Madan.
Rajiv ji, I have put my details. Please stay in touch. आप हमारी WhatsApp number इसमें है आपके पास, तो उसमें please stay in touch, क्योंकि पवन जी के साथ थोड़ा बातचीत करें।
सर, मुझे खुशी होगी सर, आप जैसे महानुभावों के साथ हम काम कर पाए तो आगे हम अपने साइंस को बढ़ा पाएंगे।
आज अपने साथ सर
आगे
इंडियन मिलिट्री से आज अपने साथ डॉक्टर देव कल्याण जी जुड़े हुए हैं, जो स्पीकर थे हमारे, और वो लेफ्टिनेंट कर्नल है और प्रोफेसर है मिलिट्री हॉस्पिटल कॉलेज के।
नमस्कार जी, नमस्कार।
प्रणाम सर।
तो इसीलिए मैंने कहा सभी का स्वागत है, बहुत बहुत धन्यवाद। अब मुलाकात होगी हमारी 19 तारीख को वर्ल्ड फोटोग्राफी डे के दिन, और उसके बाद फिर हम लोग मिलेंगे नेक्स्ट सैटरडे को जीसीपी सीरीज में। थैंक यू वेरी मच। गुड नाइट।